Nobel laureates, ladies and gentlemen, a very good afternoon to all of you. It's a pleasure and a privilege for me to welcome you all to the 2013 Nobel Lectures in Physiology or Medicine, which for the first time take place in our new Aula Medica building of Karolinska Institutet. My name is Anders Hamsten. I serve as Vice Chancellor and President of our university. The core of the Nobel Week, a particular highlight of which are the Nobel Lectures, is to celebrate science and to rejoice at the unique contributions of a number of outstanding individuals. It is therefore an honor for me to extend my warmest welcome on behalf of Karolinska Institutet to Drs. James Rothman, Randy Scheckman, and Thomas Südhof, the 2013 Nobel laureates in Physiology or Medicine. As eloquently commented upon in the last issue of the journal Cell by Susanne Pfeffer, you received the Nobel Prize for your contributions to teasing out what should be called the membrane magic. I now call upon Professor Urban Lendahl, member of the Nobel Committee, who will introduce the laureates.
Dear laureates, distinguished guests, friends and colleagues, I'm privileged and honored to stand here today to introduce the three Nobel laureates in Physiology and Medicine 2013, Dr. James Rothman, Randy Scheckman, and Thomas Südhof. Together, these three scientists have fundamentally changed our view of how cells organize the transport of molecular cargo. And they've explained, for example, how neurotransmitters and hormones can end up at the right destination at the right time. They've unraveled beautiful cellular machinery that controls vesicle docking and fusion. And without this machinery, life would indeed be pretty grim for us eukaryotes. In an evolutionary perspective, eukaryotic cells must be considered a success story. This is to a large part because of the internal compartmentalization that leads to increased efficiency in many cellular biochemical processes. There is, however, a price to be paid for this. With compartmentalization follows the need to be able to direct transport between different compartments in the cell or to the cell surface. For many substances produced in the cell and destined for export, it would simply be devastating to let them roam freely in the cell. The question of how this transport is controlled is not new. In fact, it's been occupying the minds of generations of cell biologists, including a number of earlier Nobel laureates. George Palade showed that secretory proteins are transported in vesicles from the endoplasmic reticulum to the Golgi apparatus and onwards to the cell surface. Günther Blobel demonstrated how intrinsic cues in proteins govern their localization in the cell. Otto Loewi, Henry Dale, John Eccles, Bernard Katz, Julius Axelrod, and Ulf von Euler elucidated important principles for synaptic transmission and showed that release of neurotransmitters from vesicles was indeed controlled with great precision. But despite the progress in all these areas, the question of how molecular cargo was routed lingered. What was the mechanism that stopped the vesicles with their precious cargo from getting astray in the intracellular maze? And the solution to this problem was provided by our three laureates. James Rothman took a biochemical approach and was, against the prevailing wisdom at the time, able to reconstitute vesicle transport in vitro. This system was critical for identifying molecular machinery controlling the vesicle fusion and docking. And combined with purification of key proteins from the brain, this allowed Rothman to formulate the SNARE hypothesis, a landmark concept that has aged like a good wine and has become refined over the years. Rothman's work also laid a foundation to understand how spatial precision was achieved by the specificity of cognate V and T snare combinations. Randy Scheckman took a different approach and turned to genetics. And his decision to use Baker's yeast to identify key genes in the transport process was very bold and courageous. And this journey into uncharted territory was indeed very successful. And the clever use of temperature-sensitive mutations combined with a very innovative screening approach was likewise important for success. Scheckman's work produced the genetic basis of vesicle docking and fusion and led to the identification of many of the important genes in this machinery, as well as an understanding of where in the process each gene acts. Thomas Südhof was intrigued by the mechanism for precise release of neurotransmitters at the synapse and the role of calcium in this process. Südhof's work led to the discovery of how calcium sensing was mediated and controlled. And beautiful work on synaptotagmin in mouse models, culminating in the classical 2001 Nature paper, clarified how the necessary temporal precision was achieved. And for those of us mortals who sometimes complain when reviewers and editors want a lot more data in our papers, we should take a look at the Nature 2001 paper. That, my friends, is a data-rich paper. The discoveries of Rothman, Scheckman, and Südhof are not only paradigm-shifting in basic science, but also for medicine. It's an emerging notion that mutations in the genes and the vesicle docking and fusion machinery cause disease. Similarly, the role of this machinery in key processes such as nerve cell communication, hormone secretion, and immunological responses is becoming increasingly evident. And we're presenting this year's prize. We have used various metaphors to describe these amazing discoveries. We portray the cell as a busy port in need of a logistics control center, and we liken the mutations in the yeast cells to traffic jam or collapsed public transport system. But despite our best efforts, I quite frankly don't think that we're even close to capturing the true beauty and importance of the transport system that you have unveiled. And therefore, I believe it will be much better to hear the story from the laureates themselves. So let me begin by introducing our first speaker, Dr. James Rothman. Dr. James, sorry, there's more to come here. Dr. James Rothman received his PhD from Harvard Medical School in 1976. And after a postdoc at MIT, he moved in 1978 to Stanford University, where he started his research on vesicle transport and fusion. Rothman then worked at Princeton University and Sloan Kettering Institute for Cancer Research. And he is currently professor and chairman in the Department of Cell Biology at Yale University. Please join me in welcoming Dr. Rothman, who will present his Nobel lecture under the title, The Principle of Membrane Fusion in the Cell. Please.
Yeah. Thank you. Is my presentation up yet? Thank you. Well, thank you for a wonderful introduction to the three contributors to the lectures today. It's an honor beyond measure to be able to be here today. And I will focus my lecture on the principle of membrane fusion in the cell. Now, but before I get into the substance of the science, the first question that you're always asked as a new Nobel Prize winner, in medicine it's early in October on a Monday morning, and it usually comes within 5 or 10 minutes of an awakening phone call, is how did you learn about the Nobel Prize? How did you learn that you'd received the Nobel Prize? So I thought I would present a very specific version of that in my case. In Stockholm, at a decent hour, everything is in good order. as it should be. All outcomes are known. Joran, the secretary of the Nobel Committee, Joran is well dressed and well coiffed for this very special occasion. I'm sure he doesn't always come to the lab that way, for those of you who know him in the lab. And he picks up the phone, and in some instances, though it turns out this year not in all instances, he reaches the new Nobel Prize winner. I later told Joran that he must have been an excellent practitioner of medicine when he practiced medicine, because he has such an excellent bedside manner. And of course, this was ultimately, from my perspective, if not his, a bedside conversation. Now, obviously, things were in a little bit more of a disarray in New York early that morning. The second question, I should say the first question that every new Nobel Prize winner asks him or herself, however, is different. It is, why did it happen this year? Why not 10 years ago? Why not 10 years from now? Or why at all? And it turns out, in this image is the secret. Because you'll see, next to the small set of dossiers in front of Joran, there is a much larger pile of other dossiers. And this is the secret, apparently. There are a large number of very good candidates. I imagine our dossiers were in that pile, probably for some years. And the great mystery is, how do you end up in this pile, not in that pile? I certainly have no answer to that, but speaking on behalf of myself and the other laureates this year, we really appreciate your having chosen us this year. You have many, many fine choices to make. And in all seriousness, we know that there are many prize-worthy discoveries that will never get Nobel Prizes. And we're very fortunate that our number came up this year or at all. And so we're tremendously honored. Now, the story of many of our talks actually today, and certainly the story of the work that led to my contributions, begins with George Palade. I actually happened to occupy George Palade's former office at Yale University. I'm the chair of the Department of Cell Biology that Professor Palade founded. Of course, as we heard a few moments ago, the concept of intracellular transport of proteins is classical work. It was well understood that the cell is divided into a series of membrane-bounded compartments, each with their own unique functions. And one of these compartments in particular, the endoplasmic reticulum, is studded with the particles that make proteins, ribosomes. And these ribosomes manufacture many different types of proteins, up to 10,000 different types of proteins, that need to go from where they're made to where they end up to do the jobs that they do in the cell. Now, what Palade and his coworkers discovered is a process of intracellular transport. Many of these proteins are actually physically moved from the endoplasmic reticulum to an intermediary location called the Golgi apparatus, named after an early Nobel Prize winner, early in the 20th century, Camillo Golgi, and then on to any of many destinations in the cell, and in particular to the secretory storage vesicle, which Palade and his colleagues so beautifully elucidated, that stores proteins that will be released into the blood or into the digestive tract, typically. Now, how do these proteins go from where they're made to where they end up? And how do they know where to go? These proteins we refer to as cargo. So the key question that was unanswered by Palade's generation of cell biologists is, how is the right cargo delivered to the right place in the cell at the right time? Now, they had a good idea that turned out to be correct, because they noticed there were many small spherical objects that they called vesicles. And they thought that they might be transport vesicles. They're actually very small. They're about 50 nanometers roughly in diameter. And that means they're about 500 hydrogen atoms big. That's really very, very small. But they can be packed with proteins. And Palade envisioned that they form by a process of budding, for example here, literally blebbing off of a membrane, traveling over to another membrane, and joining with it, merging the membrane of the vesicle with the membrane of the target organelle, a process that he termed membrane fusion. And so it was postulated that these vesicles might be involved as carriers of the cargo, and that there might be many types of vesicle in the cell to explain the transport of many different types of cargo to many different places. But at the time, these vesicles could only be seen in static images in the electron microscope. So this is where I entered the problem, the field, and I entered from a unique perspective. I was a child of the 1950s, and I will come to that in a minute. I've just lost my place. I meant to say, really, why is this cargo process, delivery process, important for physiology and for cell biology? And I think we've already heard of the underlying importance for cell growth and division, because the many compartments in the cell are served by these vesicles, different types of vesicles, that allow for the very propagation of the three-dimensional organization of the cytoplasm. More than this, the delivery process of vesicle fusion underlies a great reach of endocrine and exocrine physiology, as exemplified here by insulin stored in the beta cell of the pancreas before a meal, and then after a meal, insulin is discharged in order to help handle sugar in the body. And similar hormonal systems are involved in really every aspect of integrative metabolism, not to mention signaling in inflammation and immunity and many other important pathways in cell growth, development, and communication. A cell communication process of the greatest refinement occurs at the ending of every nerve, in which the electrical signal needs to be passed from one nerve to another or from nerve to muscle. And this involves a specialized vesicle called the synaptic vesicle, also discovered by Dr. Palade, that stores neurotransmitters that can then be released at the right time, already having been located to the right place, to achieve the very rapid speed of information processing that we need in the brain. So vesicle fusion serves all of these biological processes, which, as you've heard, are perturbed in a number of important chronic diseases. Now, looking at all of this complexity, it might be natural to conclude that there must be different mechanisms underlying all of this different biology, because in each of these cases, different cargoes are involved, different cell types are involved. The cargoes are released under different physiological conditions in some cases, or withheld in others. But the simplest idea would be that underlying all of this is one very basic, one fundamental mechanism. And it is the story of that discovery that I would like to principally tell you right now. So the physics of membrane fusion is not really the problem. The physics of fusing membranes, any vesicle membrane, is really no different, no more complicated, no less complicated than the coalescence of two soap bubbles, which is what you see here. A biological membrane is a lipid bilayer. It therefore consists of two lipid layers, and fusing two biological membranes to each other is literally the same as fusing two concentric soap bubbles together. The physics is not the problem. The biology, however, is the problem. How do cells harness this physics, then, to provide specificity to deliver the right cargo to the right place at the right time? So now we come to my role in the story. I was born in 1950, and I grew up in an era when physics was very much in the public eye. And I had an orientation towards quantitative thinking. I was sort of naturally inclined mathematically, and I was fortunate to grow up in a family that was focused on education and science and medicine. As a result, I couldn't remember a time as a child when I didn't want to be a physicist. And so I learned to think about the world in the way that a physicist does. I studied physics when I was a student at Yale University in college, almost until the end when I became interested in biology. But I looked at biology in the same, and I still do, in the same way that a physicist looks at physics. So what characterizes the way a physicist thinks? Physicists always seek universal laws, and they look for great simplicity. Now, one of the reasons I sometimes annoy my colleagues in biology is that I am accused of being too simple-minded, always looking for the simplest explanation. And my view, what I have found in my 40 years in science is that if you look for the simplest explanation, you're almost certain to find it. I've also found that if you look for great complexity in biology, you will also find it. So there's room for all styles. I prefer the style of Isaac Newton. An apple falls from a tree, and you have the hubris to conclude that this is also the explanation of why the moon orbits the Earth. You seek universal laws that are simple and applicable everywhere. So I've oriented my life in science towards trying to study the most fundamental things that I can, even though they appear to be diverse and complex, to try to see if there is a simple underpinning that explains a great deal. And in the area of vesicular trafficking and membrane fusion, though there is complexity, there is also certainly simplicity. Now, the simplest hypothesis to explain the intracellular transport pathway that was discovered by Palade is actually that there is intrinsic chemical specificity in the vesicles. That, however, was not the prevailing mood when I undertook this problem. I first learned about George's work when I was a first-year medical student at Harvard, and I was amazed that this type of complexity could occur in a cell, and I wondered how it could occur. The prevailing wisdom was that anatomy dictates specificity. In other words, here's a classic view in which the endoplasmic reticulum is juxtaposed directly to the Golgi apparatus, and in between are many teeming vesicles seemingly on their way from the endoplasmic reticulum to the Golgi. It's as if they were perfectly placed there to go no place but the Golgi. And so when I said I wanted to study this and identify these proteins in cell-free reactions in the way that a biochemist would, the senior people in the field all told me that I was destined to fail because the anatomy was so important, which provides my first lesson for those of you who are students here today, which is in itself an oxymoron. Never listen to older scientists. What I did instead was what I always do. I create a model before knowing any facts whatsoever about how it might work. So this is a model I drew in the late 1970s. You see it was published in the early 1980s. And frankly, it looks uncannily like the diagrams today of the vesicle trafficking pathway because it is so simple. You need to have a mechanism to form the vesicle, proteins that target the vesicle, and if you have this type of intrinsic specificity, then the anatomy will follow from the action of this system. That was my hypothesis. And so I set out to develop a technology and an approach that would allow the proteins underlying this intrinsic specificity to be discovered. And the remarkable prediction of this simple hypothesis is that vesicle traffic, which almost paradoxically itself generates the anatomy in the cell, can nonetheless occur in the absence of any pre-existing cellular organization, in other words, in a cell-free extract. Now, obviously, we were not the first to think that to take the approach of studying a biological process in a cell-free system. Actually, the first person to have discovered is Eduard Buchner, who won the Nobel Prize in 1907 for his discovery that the production of alcohol, fermentation of sugar, thought at the time, as a result of Pasteur's study, that all life comes from life, using yeast cells. At the time, it was thought that essential life processes, like fermentation, like biological oxidations, could only occur in living cells. Buchner discovered otherwise, and that dispelled firmly the concept of vitalism. It actually founded the field of biochemistry, and it led to the belief that he first enunciated that all of life is the result of chemistry. Nobel lecture. Really quite a remarkable statement. 1907, we are seeing cells more and more clearly as chemical factories where products, that is proteins and others, and chemicals, are manufactured in separate workshops. That was even before the compartments were known, guided by enzymes acting as the overseers. Really remarkable foresight. Now, scientists following this insight have succeeded in reproducing entire metabolic pathways. Then they succeeded in manufacturing of enzymes, the discovery of how chromosomes are duplicated. More recently, in our field, the discovery by Günter Blobel that proteins can be moved across membranes in cell-free systems. And then we took on the challenge of asking whether the three-dimensional organization of a cell, an even more complex process, could be reproduced in the test tube. Now, in order to do this, and a major theme of my lecture today really is how is a discovery made, okay? And I think you'll conclude with me by the end of the lecture that it's a complex set of circumstances that lead to original work in science, whether it be this body of work recognized today from my hands or from that of my other colleagues or in any field. It's a complex ecosystem that we operate in, and there are many factors that enter to produce the ultimate success, which include factors that are good luck as well as insight. One of the great fortunes in my life was to be invited as a young man at the age of 25 to join the biochemistry department at Stanford University, which was then still very much under the guidance of Arthur Kornberg. Arthur was the first person to reproduce chromosomal synthesis in a test tube. He discovered the first polymerase that makes DNA and the basic principle that's involved. And he was certainly the master of enzymology in his time. Enzymology is what the descendants of Buchner were set at that time to practice, the use of enzyme chemistry and enzyme fractionation to discover biochemical mechanisms by delineating pathways. And so having the good fortune to set my own laboratory up under the influence of this master on a day-to-day basis was an extraordinary thing. But that's not enough. You need to find the right colleague, which in this case was Erik Fries, a Swedish gentleman who is now at the University of Uppsala, and I'm hoping is here in the room today. Erik, are you here? Would you please stand up so we can applaud you for what you've done. Thank you, Erik, and I look forward to seeing you after some years, right after the lecture. This is Erik around 1980, and this is actually the gift that I gave him, which will be donated shortly to the Nobel Museum. And this is the homogenizer that was used by Erik to first reconstitute intracellular transport in a cell-free extract. It goes by the name more Dounce per ounce, named after the Dounce homogenizer. So Erik, I'm hopeful that you brought that with you today, and I thank you for being willing to give up what I think has been a cherished possession, and I know has been in good hands all these years. Well, even the right person in the right environment isn't quite enough, because you then have to get into the lab and you have to work hard, and there was a lot of hard work until we found the right method. And again, this is now very old work, and I won't go into it in detail, but just to say that the key idea that allowed us to measure reconstitution of transport was to follow a cargo molecule called the VSV glycoprotein that is passing through the endoplasmic reticulum in the Golgi on the way to the cell surface. It happens to be a sugar-laden glycoprotein, and within the Golgi apparatus, the sugars are elaborated. And what we measured was the elaboration of these sugars onto this protein as it starts out in the membrane system, but now in a cell-free extract. And we measured the addition of further sugar residues onto this protein, which signaled the fact that this membrane protein must have moved from one membrane compartment of Palade to another. Now, in order to see that process selectively, we came upon a scheme where we actually mixed extracts containing membranes from two different types of cells. One was a mutant fibroblast that lacked a particular sugar transferase. The other had a good copy of the sugar transferase. The extract lacking the sugar transferring enzyme, that is the one that had the cargo protein. The membranes from the other type of cell, they didn't have the cargo protein. They were not infected with the virus, but they had a good copy of the sugar transferase. So the only way in which this cargo could get modified by that sugar is if it could move from the membrane originally present in one cell to the correct membrane in a membrane system originally present in a different cell. And so by definition, success in this measurement means that no pre-existing cellular organization could contribute, which is what we wanted to test. That was the essence of the intrinsic hypothesis. It also implied that there would have to be a diffusible carrier, a vesicle, in other words, that would move the cargo between these membranes. Well, after some time, in fact, on May 10th, 1979, success was had. And this is a gel, an autoradiograph of a gel. You can see a tiny, tiny little band right here that's barely visible. But that, Eric and I found electrifying because it meant that even a tiny amount of this VSVG protein was being modified as a result of transfer in the cell-free extract. Well, over the next several years, William Balch especially joined the lab, and we improved this assay so that we could extract a very large signal as compared to a very small background, and therefore make this a useful assay for enzymology and enzyme purification. And that was done by radioactive tagging of the sugar so that we could then directly monitor the incorporation of the sugar into this protein, silencing, in effect, the background of all of the other copies of the G protein that were present in the extract. And that was a very beautiful assay. It showed a requirement for cytosol, which meant there must be enzymes of some sort that help the vesicles get made, and perhaps other enzymes that help them fuse. ATP was required. You see, if you omit it, very little transport, so there must be an energy requirement. And so we set about trying to understand how this process works. In our mind, this was a very fancy version of a biochemical pathway, where you have a substrate, a product of that product, and another product. But instead of being small chemicals, these intermediates were vesicles at various stages of forming and fusing. And we knew that it was necessary to turn to electron microscopy to visualize these intermediates. And in order to do that, we began a collaboration with one of the great electron microscopists of our time, Professor Lelio Orci of the University of Geneva. And this allowed us to monitor the cell-free incubations to see if indeed they produced vesicles, and indeed if these vesicles would fuse. In particular, as in many past successes in biochemistry, the use of specific chemicals that inhibit different stages of the process were vital to us in dissecting. It turned out that non-hydrolyzable GTP analogs prevent the fusion of the vesicles, as do low concentrations of a sulfhydryl alkylating reagent, N-ethylmaleimide, but they accumulate vesicles at different stages. We got this idea from the classic studies of biochemists who, like Ephraim Racker and Britton Chance, who discovered the mechanism of oxidative phosphorylation. And they they used specific poisons to block specific stages in the electron chain, a transport chain. We found that both of these inhibitors, when we looked in the electron microscope, accumulate vesicles. With non-hydrolyzable GTP, these vesicles accumulated with something that had not been seen before in this context, an outer coat, which turns out to be made of protein. And it allowed us, for the first time, to purify transport vesicles. And what you see here is an early success of the laboratory, especially Vivek Malhotra in the laboratory, two at the time, obviously, in which he isolated transport vesicles that turned out to be functional carriers. They could be produced from membranes in one incubation, added to a second incubation, and shown to go on to fuse. So these were functional transport vesicles, no longer seen just in the electron microscope, but now isolated in pure form as functional entities, so we could understand more about how they bud and how they fuse. This led, in fact, to the discovery of the coat proteins, and we could then separately reproduce the budding process from isolated membranes, and even eventually from lipid vesicles. And in the process, we discovered the very basic GTP-switch mechanism in which GTP is bound to a G protein called ARF, which then recruits a coat subunit, coatomer, now called COP1, assembling into a spherical shape that templates the membrane. That's the coat that you saw in the electron microscope. And once the vesicle has formed, GTP is hydrolyzed, and that releases the coat so as to release the enclosed vesicle to enable it to fuse and to allow the coat protein to be reused again in a round of vesicle budding. And I'm sure in Professor Scheckman's lecture, you'll be hearing much more detail about how this process works. But what I would like to do now is turn to the inhibition by N-ethylmaleimide, because this led us to the very heart of the process of membrane fusion and the solution to the problem of the right place at the right time. Speaking of time, I better check my watch. Now, when you look in the electron microscope, you see vesicle after N-ethylmaleimide treatment, you once again will see vesicles accumulating that fail to fuse. They are lined up trying to fuse, apparently, in this case with the Golgi membrane. But they lack the coats. Why? Because the coat has already come off, but now they fail to fuse. So there must be an N-ethylmaleimide sensitive factor, or NSF as we called it, that is needed, as again Malhotra discovered with us, for the process of membrane fusion. This protein was purified as a soluble enzyme from the cytosol by Bloch and Felix Wieland, again still back at Stanford. And so what we now had was a pure protein, purified in a classic manner as an enzymologist would, which when added back to an NEM-poisoned cell-free reaction would reconstitute transport and therefore reconstitute fusion. Therefore, this protein, NSF, must be needed for membrane fusion in some way. We soon found that NSF hydrolyzes ATP, which explains to a large degree the ATP requirement that we saw early on. But we were left with a mystery. NSF is a water-soluble ATPase. It actually does not bind to the membranes, certainly not the Golgi membrane. How can a protein which doesn't even bind to a membrane engage in fusing membranes? We went back to the cytosol and imagined that there must be a soluble NSF attachment protein, which we called SNAP protein. And the reason for this is we found that NSF would bind to the membrane, but only when we added back cytosol free of NSF. So there must be a SNAP protein. We were able to purify that protein according to the assay that I just mentioned. Now we have two pure proteins. Both are water soluble, both from the cytosol. Both are required for vesicle fusion. SNAP and NSF. We found that unlike NSF, the SNAP protein would bind to the membrane independent of NSF. And it would bind in a saturable fashion to the membrane. And this meant there had to be receptors in the membrane for the SNAP protein, which was required for fusion and required to bind the NSF ATPase. I'm now taking you up to 1993. So I'm hoping you're beginning to think that we're getting closer to the process of how the membranes fuse. Now, it turns out that NSF and SNAP proteins are used for every fusion reaction that was known at the time in the cell, many, many fusion reactions. They therefore, neither one can provide specificity. And you remember that the cell has to have many different types of vesicles. Each one has to fuse with the right place. So if NSF and SNAP are not providing the specificity, but instead are machinery helping in one or another way, facilitate the fusion reaction, specificity has to come from someplace else. Where better than from the membrane itself, which is already endowed with the identity of being an endoplasmic reticulum, or an endosome, or a plasma membrane. So we therefore sought the SNAP receptor, thinking that it may be involved in membrane fusion, and in particular that there may be many different types of SNAP receptors, each in its own membrane. We abbreviate SNAP receptor as SNARE, S-N-A-R-E. This is where the word SNARE comes from. Okay, so the next step was to isolate the SNARE proteins. Now, again, you need the right colleague. In this case, Thomas Zollner joined the laboratory, having just arrived from Walter Neupert's laboratory in Munich, and being an extraordinarily well-trained biochemist as well as a brilliant young scientist. He is currently a professor at the University of Heidelberg and has promised me that he would actually show up today, despite the fact that I warned him I would ask him to stand. Thomas, are you here? I hope. Would you please stand up? So this is Thomas Harold Zollner, who was the first person to discover the SNARE complex in my laboratory. And he developed a method that was very intricate and took advantage of the intrinsic enzyme properties of NSF. When Thomas isolated these proteins from the extracts of brain, he discovered something quite remarkable. Now, I want to just take a moment to point out that we were not working with brain, you see. We had no ambitions to contribute to the understanding of neurotransmission. This is the wonderful thing about basic science. I don't think Eduard Buchner was necessarily researching yeast cell-free extracts because he wanted to brew better beer, although I suspect that's why his work was paid for, okay? And this comes to the nature of basic science. We used brain extracts because measurements in the lab suggested that the concentration of SNAP receptors was 10 times greater per gram of total protein in the brain than in any other tissue in the body or in any cell line. And the reason for that, in retrospect, is that we are so active in membrane fusion because our synapses are firing all the time, okay? I personally wanted to become a neuroscientist. I never wanted to become a biochemist. Actually, when I was a medical student at Harvard, I applied to study in the biochemistry department and into the neurobiology department. And I was rejected by the neurobiology department, though I wanted to study synaptic transmission. As a result, I went to my second choice department, and I had the great privilege of learning biochemistry with Eugene P. Kennedy, and I became a biochemist. But it just goes to show you, destiny speaks for itself, and you never know where life will lead you, and the very fact that we chose brain is the reason that we were not only successful in this experiment, but we simultaneously were able to explain the core mechanism of neurotransmitter release. And what we found in one experiment that was extraordinarily exciting in February of 1993, when we went fishing with NSF and SNAP protein, is that we pulled out 1, 2, 3 other proteins identified by microsequencing that were present in stoichiometric amounts. And what was utterly amazing is that each of these SNAP receptors, as we call them, or SNAREs, was an already known protein. It was known from the work of a number of laboratories who had been intentionally trying to approach synaptic transmission by sequencing many dozens of different proteins that are associated anatomically with the synapse, and in particular, the synaptic vesicle. But we didn't know which, if any of these, was directly involved in membrane fusion, or if they were involved at all. But when Thomas found a complex of one of them, called VAMP or synaptobrevin, known to be in the synaptic vesicle, with syntaxin and SNAP25, which were located in the plasma membrane with which this vesicle fuses to release neurotransmitter. At that very moment, it became apparent to us, though it took some years to really prove, that a molecular complex, which we called the SNARE complex, we're calling these proteins SNARE proteins, would be vitally important in providing for fusion in association in one or another way with NSF and SNAP and ATP hydrolysis by NSF. We went on to propose the SNARE hypothesis, that the cell has many homologs of VAMP, which we call the V-SNARE because it's in the vesicle, and syntaxin and SNAP25, which we call T-SNAREs because they're in the target membrane in this case. And we imagined each membrane vesicle and each target membrane would have its own unique, similar V and T-SNARE that would partner with each other specifically, both initiating the process of membrane fusion and at the same time in a kind of molecular two-for-one, providing specificity. Perhaps not the only source of specificity, but a critical source of specificity. This very simple hypothesis was validated a few years later when we could take these proteins from the synapse as pure proteins and put them into artificial lipid vesicles, and we found that no proteins other than these proteins were necessary for these vesicles to fuse. The ultimate version of this experiment, though, and the one that I'm most proud of, and what actually represents about 20 person-years of work, is summarized in this slide. And what we did was, thanks to the work of Randy and his colleagues and his students, all of the SNARE genes in yeast were ultimately known. And we were able to express each of the SNARE genes from the yeast genome one at a time and put them one by one or in combinations 2, 3, 4 at a time in literally hundreds of different combinations in different liposomes to see which of the hundreds of possible combinations of liposomes are inherently capable of fusing. How much specificity of fusion is actually encoded in the SNARE proteins. And out of the hundreds of possible combinations in yeast, only a handful are actually capable of fusing, and the others not at all. And what that means is that specificity of compartments is encoded in these proteins. A physicist looking at this and getting this result would be able to immediately map out exactly what transport pathways can and do occur in the yeast cell. That is how predictive this hypothesis was. So to summarize then, our current knowledge of membrane fusion. SNARE proteins assemble in a 4-helix bundle, as it was discovered by X-ray crystallography, in particular by Brunger and by Jan. And they assemble by a process of zippering in which this 4-helix bundle forms between two membranes. The zippering process produces an inner-directed force that brings together the two membranes, and it results eventually, just like the two soap bubbles, in their physical coalescence. The role of NSF turns out to be to disrupt the SNARE complex by hydrolyzing ATP using SNAP protein as an accessory, allowing the SNARE complex to be pumped back up to its high energy state in which the molecules are dispersed and largely unfolded. The energy of protein folding released during the assembly of the SNARE complex into the 4-helix bundle would release energy, and that energy released from protein folding ultimately is what drives bilayer fusion. This next series of slides illustrates this somewhat more specifically, and here we see the assembly of the V-SNARE from an unfolded state into the 3-helical bundle T-SNARE, pulling the membranes together, creating an inner-directed force which actually raises the water pressure, we believe, the hydrostatic pressure between the two bilayers, as Fred Poncet and I have estimated, into the range of several hundred times to 1,000 times the pressure of the atmosphere. Under these atmospheric pressure conditions, it is well understood that lipid bilayers decompose into unstable structures, just like the fusing soap bubbles, and that under the continuing force until the assembly of the SNAREs is complete, the membranes are forced together, we believe, in a manner like this. It's not fully understood yet, to open a fusion pore that releases the neurotransmitter, or in general, merges the vesicle with the target membrane to allow content to be distributed. Now, for those of you who are non-scientists, I want to illustrate this with a hairpin and two bubbles. If you watch this video, you will see a lady's hairpin. Everybody who has had the experience of opening a hairpin. Just like opening a snare pin, it takes a lot of energy and work to do this. Now here what we're doing is we're taking the two ends of the open snare pin, it's like unfolding the snare pin, and we are inserting these into two balls. Now we let go of the two balls, and what will happen is the two balls will be pushed together as the snare pin assembles, forcing the two together, and now an inner-directed force will push these two bubbles together, these two vesicles together. If the force is strong enough to overcome the surface tension of the bubbles, if they're rubber balls, it will not be. But if they're biological membranes, it can be, and when it is, these are coalesced together. It's simple physics. The magic of the biology is in the pin, because the snare pin, unlike this hairpin, is naturally in two parts, V and T. This hairpin comes in one part, but in the cell, each V snare needs to find its T snare, and only then is this, when the right combination is made, is this pin allowed to assemble to force the vesicle into its target. Now, I want to quickly emphasize that this is the core principle of membrane fusion. There are many proteins that regulate this process and add specificity, but it is also not a massive oversimplification. Here, nor is it fanciful. Here, within the last year or two, with my colleague at Yale, Yongli Zhang, and our colleagues, we've actually taken a single snare complex, a single snare pin, and positioned it between two balls. These are two beads that are entrained by light, optical tweezers. And here we manipulate the single molecule to pull the last part of the zipper apart. And then we watch this last part of the zipper come unzipped, rezip, unzip, rezip, and unzip, rezip. And what it is a binary switch. It's an all or none transition that represents the final step in membrane fusion. So this is not fanciful, it is actually physics. And from the rate of this rezippering, it occurs way faster than a millisecond. Now, the beauty of this physics is it creates a platform for regulation, because a major issue, and the one that we work on now, as do many other laboratories, is how is it that neurotransmitter can be released within a fraction of a millisecond of the entry of calcium into the nerve ending and the recognition of calcium by the mechanism that Tom Südhof discovered and no doubt will describe today. And the answer, in broad terms, is that the snare complex has already assembled and it is all zippered except for this last little bit. And something must be there to prevent that last zippering in order to prevent the neurotransmitter being released until just the right time has come. It's at the right place, but it needs the right time when the nervous impulse comes. And we're gaining insights now into how that works. The two molecules that Tom discovered, and we'll tell you about, Complexin and synaptotagmin, are central to this. And we've learned in the last couple years, especially from work that we've done in collaboration with the Yale crystallographer, Karen Reinisch, and her collaborators, that the molecule complexin acts as a molecular clamp. It literally inserts itself in between snare complexes so that it gets in the way of the zippering of this last little bit of the V snare. You see, the V snare here would like to continue zippering, but it can't because complexin, like the old parable, the fairy tale of the finger in the dike, this complexin is the finger in the dike that prevents neurotransmitter from being released. It also serves, because it bridges snare complexes, to organize the snares into a supramolecular complex that forms a kind of a zigzag array that makes this into a cooperative structure that we believe explains, but this is not yet entirely proven, the very high degree of synchronicity, and which is so necessary for synchronous synaptic transmission. This isn't all of the story. Active work is continuing, and our current directions include trying to understand how synaptotagmin, the calcium sensor, which is also vitally important for clamping and regulating the release, how this protein is organized. And very recently, Charles Sindelar, a biophysicist in the biophysics department at Yale, and I and our colleagues have found that synaptotagmin self-organizes into beautiful rings on the surface of the membrane, and it is our belief, but a speculation, that these rings serve to organize, to radially organize the zigzag array and further coordinate the production of a three-dimensional molecular machine which we're only beginning to understand. It turns out that rings in vesicles have become a theme of my life. Another project that we're working on is better designing vesicles by using rings of DNA origami as templates to make vesicles of absolutely uniform size and predetermined content, so we can exactly determine by engineering, nanoengineering, the number of protein molecules in a vesicle and their orientations. And this facilitates many things, not the least being the studies of the fusion process itself. And finally, in collaboration with a number of other colleagues, faculty colleagues at Yale, and with the very generous support of the Wellcome Trust, we're able to develop super-resolution optical microscopy to new levels of speed and resolution, constricting the ring of resolution to the point where we expect within the next 2 or 3 years to visualize in real time and with 10 or 15 nanometers spatial resolution, better than a vesicle. We expect to see individual vesicles moving in the cell and be able to read out the content and cargo and machinery they contain to answer long-standing questions that remain about the organization and dynamics of the compartments in the cell. Now, I want to conclude by coming back to my original theme about what makes for the possibility of a great discovery in science. And I hope you have come to the conclusion that I have, that yes, the individual scientists contribute, and sadly, only some of us can be recognized on platforms such as the one I'm on today. However, it takes more than that. It actually is a complex ecosystem. And this was first pointed out in an elegant article by Hans Krebs, who is famous, of course, for the Krebs cycle, the TCA cycle that many of you have learned about and memorized. It actually was discovered by somebody. And it was a brilliant piece of biochemistry. You'll see that Krebs, as he points out, in The Making of a Scientist, was taught by Otto Warburg, who discovered the basic principles of biological oxidation, and who worked for Emil Fischer and was trained by him, as was the famous Eduard Buchner, and so on. In fact, all of these individuals were descendants of the 19th century German dye chemist von Baer, who was interested in isolating natural product dyes and learning how to make better dyes, which, by the way, gave rise to important branches of organic chemistry. This was a 50-year ecosystem that led to the development of modern biochemistry, and I have to say, includes me, in the sense that I was a student of Eugene Kennedy, and I learned enzymology at his hands, and I very strongly came under the influence, as others who you will hear from today, and essentially was a pupil of Arthur Kornberg. So I fall into the same lineage, and the work I did very much, as I hope I've emphasized, was influenced by this way of thinking, beginning certainly with Buchner. Now, the importance of this is that these ecosystems are fragile. We saw this with the rise of Adolf Hitler at in the late 1930s. It's no accident that all of this biochemistry occurred in and around Germany, because that's where it developed. And Germany and Germanic Europe were, without a doubt, this epicenter, not just in physics, or chemistry, or biology, but also medicine. That all changed. And those who survived emigrated, many to the United States. As a result, many of your Nobel laureates, much of the science that's important in all of these fields has been done in the United States. That culture was grafted onto the United States with our entrepreneurialism and the cultural preparedness to fail, and I think was enriched. Now that culture stands deeply threatened by brutal cuts in support for basic research, and it can go away. It may take 50 years to fully establish. Actually, European science has been rebuilt. It is now as vital, as you know, as American science. But it took a full 50 years for that to happen. So there we are, and I hope that we will not allow this to happen in the United States, but we are under some threat. I'd like finally to turn to some inspirational words from Buchner, who ends his Nobel lecture with a lesson for all of us, and I share this with especially the students, because you need to understand when you hear a Nobel lecture about old work from old people that there remains much to be done, but you can also learn from your elders, as I learned from Edward Buchner. And you must never think that there are, that a day will dawn in which optimism should not prevail. And in his view, we should have every confidence that the day will dawn when even those processes of life that are still a puzzle today will be understood by us scientists, and indeed understood in chemical terms. So I have many people to thank, and there are many dozens of contributors who have helped me in my lifelong journey with bubbles, an example of which you see here. Bubbles are not only educational and important for all of life, they're also fun. And I can't possibly acknowledge each of the individual contributors, but I would like to ask some of the contributors to my personal life to stand up. Shouldn't choke up. Most importantly, Joy Hirsch, my wife. Please. Who has been my partner in life, as well as my intellectual partner, and to whom I owe an awful lot. I'd like to ask the members of my family who are here today to stand up. I'm hoping you're here. I haven't seen you yet today. But you add and enrich our lives tremendously, and we're grateful for you. And finally, the many guests that we have who have been my scientific colleagues, my supporters, my friends. I think you know who you are. I'm going to ask all of you to stand up. Please. Don't be modest. Now's a good time. This is my other ecosystem, and this ecosystem actually goes back to include my roommates from college. So I thank each and every one of you today for your attention, and again, I'd like to thank the Nobel Committee for honoring our field and for honoring these discoveries in particular. Thank you. Thank you. Thank you. Thank you.
So, after thanking Dr. James Rothman, I would now like to introduce our next laureate, Dr. Randy Scheckman. Dr. Randy Scheckman studied at the University of California in Los Angeles at Stanford University, where he received his PhD in 1974. In 1976, Dr. Scheckman joined the faculty of University of California at Berkeley, where he is currently professor in the Department of Molecular and Cell Biology. Scheckman is also an investigator at the Hughes Medical Institute. Please join me in welcoming Dr. Scheckman, who will present his Nobel lecture under the title, Genetic and Biochemical Dissection of the Secretory Pathway. Please.
Well, thank you very much for that wonderful introduction and to this delightful audience. I learned yesterday that over 17,000 people have signed up to watch this lecture live around the world. And then, of course, the lectures will be posted at the Nobel Prize site and be available to be viewed in the future by anyone. So to all of you here today before us, and to all of my friends around the world, and especially to my friends back in California, some of whom have awakened at 4 in the morning to watch this lecture. I salute you. Welcome all. I am particularly grateful to the Karolinska Institute, to the members of the Nobel Committee, to the Nobel Organization, for allowing me to share the stage with these wonderful scholars, James Rothman and Tom Südhof. This is a dream come true for me, and I can assure you that not only am I thrilled, but my 86-year-old father, who is here today, has been reminding me every year in early October that he was not going to live forever. And so, Dad, where are you? There's my dad. Congratulations, Dad, you made it. Well, we begin a journey. I'd like to start by telling you the origins of our work at Berkeley in using genetics and biochemistry to dissect the secretory pathway. But before I do, of course, I must start by giving credit to the pioneers who initiated the study of membrane transport in eukaryotic cells. And you've already heard the pioneer, the true pioneer, is George Palade. But I must also say, so origins and influences, I must also tell you that it was not only Palade, but his colleague then at the Rockefeller University, Keith Porter, who perfected the technique of transmission electron microscopy in evaluating thin slices through tissue, pancreatic tissue, particularly the pancreatic acinar cell, and we're able to first visualize the structures that mediate protein secretion. It was Palade's genius to recognize that small particles in the cytoplasm are the site of biosynthesis of protein molecules. Palade was the first to describe and characterize the role of these particles in protein biosynthesis, but that's only one of his many discoveries. He used electron microscopy and cell fractionation to isolate this organelle, the rough endoplasmic reticulum, and to demonstrate in the pancreatic acinar cell that newly synthesized proteins are assembled and transported across a membrane into the lumen of the organelle, the first stage in the biosynthesis of secretory proteins. But even then, that wasn't enough, because Palade, in collaboration with his laboratory, principally Jim Jameson and Lucien Caro, developed a brilliant approach to map temporally the movement of molecules as they march from one station to the next within the secretory pathway. And that is summarized, well, here, of course, is George. In this case now, more senior, he was, at this point, a professor of cell biology at University of California, San Diego. What Palade did was to map the temporal sequence of events from the endoplasmic reticulum through this structure, the Golgi apparatus, and onto mature secretory granules, which discharge their content by docking and fusion at the cell surface. His experiments were the first to appreciate that this mystical structure, the Golgi apparatus, may have at least a temporal role in organizing the flow of molecules within a cell. His discovery is no less important than the discovery of, in the 17th century by Harvey of the human circulatory system. Palade was, in a sense, the father of the intracellular circulatory system. I first learned of Palade's work when I was a graduate student at Stanford, but most importantly, when I watched a lecture of his when I was a beginning postdoctoral fellow, when Palade returned from Stockholm in 1974 to tell the American Society for Cell Biology about his work. It was a thrilling occasion for me to see not only the scholarship, but how he was revered by people in the field. And yet, as I watched him describe the movement of molecules, I, having been trained as a biochemist, was left wanting to learn more about how this process was organized. How was it that proteins could actually enter across a membrane of the endoplasmic reticulum? These proteins were water-loving hydrophilic molecules. They had to cross a hydrophobic barrier. How did that happen? How did these vesicles form? How did they know which molecules to carry en route to the structure, and so on, through the pathway? In other words, what mechanistically allowed this complex series of events to proceed? How to do this? Well, at the time, I was a young pup, just beginning an interest in microbiology. Here's a science fair project that I was engaged in 9th grade. I had, at the time, a microscope that I had purchased with earnings from mowing lawns and babysitting. I spent my entire youth in my room peering through this microscope, watching little bugs swimming around from pond scum. That microscope now sits in your Nobel Museum. I deposited it yesterday. So I was drawn to understanding how life could be experienced and expressed in the microbial world. And when I started university at UCLA, I pursued my interests in microbiology through the study of small bacteriophage DNA replication, how these chromosomes are duplicated. Because of an interest in chromosome replication, I was drawn inevitably to the pioneer, the real power in the world at the time in dissecting the process of DNA replication, and you've heard about this from Jim. His name is Arthur Kornberg, a former laureate in 1959. Kornberg was a master enzymologist. Here is a picture of Arthur at my wedding to my lovely wife, Nancy, 40 years ago this year. You can see that we were flower children of the time, but I can assure you that Arthur was not a flower child. He was a very serious scientist with the highest of standards, not only of himself, but certainly of those in his laboratory. And I learned a great deal about how to tackle a difficult problem. I was drawn to Kornberg by work that he had conducted even when I was an undergraduate at UCLA that culminated in a really quite remarkable demonstration that a small chromosome could be copied in the test tube with the enzyme that he discovered, DNA polymerase. 2 crucial papers were published in 1967 in the Proceedings of the National Academy of Sciences, shown here. In this work, Kornberg and his colleagues took a simple viral DNA template, and with the addition of his enzyme, DNA polymerase, he was able in successive stages to make copies of the single-strand template and to produce an infectious DNA molecule. Really remarkable for the time to be able to demonstrate that a single enzyme would faithfully copy a DNA molecule to produce a productive infection. So every bit of evidence from his considerable armamentarium convinced us at the time that this surely was the enzyme responsible for chromosome replication. And yet, 2 years later, in a discovery that rocked the field of DNA replication, John Cairns at Cold Spring Harbor, using a very different logic, was able to obtain a mutant E. coli in a screen of randomly selected mutagenized bacteria. He identified a mutation that killed the DNA polymerase enzyme. It was a terminating mutation that eliminated that enzyme, and yet, as you see from the abstract of this paper, these bacteria grew essentially nearly normally, and yet they demonstrated a defect in the DNA repair process. So I use these 2 examples to bracket the knowledge that one can obtain for the investigation of complex processes. On the one hand, biochemistry is enormously powerful in getting to molecular, indeed atomic resolution, but one also needs physiologic criteria, as evidenced here in the form of genetics, to define the crucial events. And as a result of Karen's discovery, it became possible to return to a lysate of E. coli, and Bill Wickner and I, as students in Arthur's lab, were able to dissect the replication process and to identify the appropriate enzymes that were actually involved in chromosome duplication. During my graduate career, as we were busy performing this enzymologic dissection, I became aware of another very brilliant series of studies conducted by Leland Hartwell and his colleagues, Hartwell also subsequently a Nobel laureate, using the simple eukaryote, Saccharomyces cerevisiae, baker's yeast. He succeeded in dissecting the pathway of cell division. The simplest approach. One simply took yeast cells, shown in this panel, which grow by a budding mechanism, and he exposed these cells to a chemical mutagen, and he looked for those genes that are essential for progression of the cell division event by collecting clones that could no longer grow and divide at a high temperature, a so-called restrictive temperature, thus identifying essential genes. And he was rewarded in this simple and elegant approach with a number of so-called cell division cycle mutants, each of which, as you see in these panels, arrests at a unique terminal morphology. That is, that the cell division cycle progresses until arrested at a restriction point where the cell can no longer progress because it's missing an essential gene product. Well, this was a powerful lesson to me. Having been trained as an enzymologist, one could rely on genetics to try to dissect a complex process. And so, as my career developed, I turned my interest after my graduate work to the study of protein secretion and protein traffic, first through a postdoctoral period at the University of California, San Diego. Under the leadership of S.J. Singer, I learned about biological membranes and how they may be put together. But I decided when I had my first independent academic career to begin anew. And in 1976, I had the great good fortune to be appointed an assistant professor in the biochemistry department at UC Berkeley, under the leadership at the time of a wonderful man by the name of Daniel Koshland. At Berkeley, I decided to take a gamble and to have a look, using the logic of Lee Hartwell, with my background in biochemistry, at the mechanism of protein secretion in baker's yeast, an organism that had been studied in many respects, but very little was known about how proteins may be secreted. In fact, if you look inside a yeast cell using the technique of transmission electron microscopy, it's not at all evident that the cell would behave at all like a pancreatic acinar cell. We'll contrast the image you see here with that of the pancreatic acinar cell. The yeast cell appears to be relatively devoid of organelles, with the exception of a background stain that consists largely of ribosomes. Nonetheless, particularly if the cell comes equipped with these nice little labels, you can see organelles, the nucleus, the endoplasmic reticulum, bits of the Golgi apparatus, but my attention was drawn initially to a collection of small membranes, vesicles, that congregate under the bud portion of the growing cell. This is the position, the part of the cell that grows during the 2 hours of growth that lead to the production of a daughter cell. And we imagined that these vesicles would convey secretory proteins to the cell surface. In my first year as an assistant professor, I had the other great good fortune to have a brilliant student join my lab, Peter Novick. First, I had to deal with controversy on the Berkeley campus. Berkeley, of course, is the home of protest in the United States, and though I thought I was safe working on yeast as a model organism, nothing is safe at Berkeley because, after all, yeast have feelings too. Well, undaunted by this opposition, Novick, who spent all of his time in the laboratory anyway, began an exploration of the process of secretion using genetics. Now, I'd like to digress at this point to introduce not only Peter, who's here this afternoon, but his wife, Susan Farrow Novick, also a graduate student in the lab, also working on the genetics of secretion. So if I could have both Peter and Susan stand and receive your recognition. One of the happy features of my laboratory is that these two were the first married couple to form that union in my laboratory, the first of 7 such marriages just within my lab. I did have one divorce, but just the one divorce. Well, Peter and I set ourselves a goal to try to dissect the process of secretion using genetics. And our idea, our crucial idea, I would say, was that these vesicles would not merely be the means by which secreted proteins are conveyed to the cell surface, but that the membrane that surrounds the vesicle would also become the building block of the plasma membrane of the cell. So you would proceed not only to secrete molecules, but to enlarge the cell using the same building block. Now, of course, any mutation that would interrupt the growth of the cell surface would surely kill the cell. And so we made the crucial prediction, as Hartwell had done, that we would have to go after temperature-sensitive lethal mutations. And we did so. After some trial and error, Peter realized that it might be possible just to obtain a mutation blocked in secretion, as Hartwell had done, by surveying a random assortment of temperature-sensitive clones. And one of the great moments in my scientific career was when Peter, having isolated this mutation called SEC1, called me down to the electron microscope to see what he saw on the screen in the stage of the EM of the electron beam. And that was an image of a cell, a yeast cell, that had never been seen before, a cell which, in this case, accumulated many vesicles. Recall the previous slide where only a few vesicles were found. In this mutant, SEC1, growing at a restrictive temperature under conditions where the cell was about to die, instead became essentially constipated with vesicles from one end of the cell to the other. This mutant, when restored to the permissive temperature, allows secretion to continue, and the content of these vesicles are discharged by release at the cell surface. So this was a wonderful discovery, and it suggested immediately to me that this was a pathway that could be probed by genetics, and so we then, over the next year, year and a half, continued to look for more mutants, but we found only a few until Peter realized, by close inspection of these cells, that at the restrictive temperature, all this material continues to be made, and yet the cell doesn't enlarge. Therefore, more mass must accumulate in the same volume, and we predicted that these cells would increase their buoyant density as the cells were incubated under conditions where they inevitably would die. And so in another really brilliant experiment, Novick took the SEC1 mutant cell and mixed it with a large excess of wild-type cells, incubated the mixture in a flask at the high temperature, body temperature, 37 degrees, under conditions where the SEC mutant would begin to die, but normal cells would progress. And then he took the mixture of cells and centrifuged it on a density gradient set up so as to retain wild-type cells near the top of the tube at a low buoyant density, whereas mutant cells, SEC1 mutant cells, formed at a dense layer near the bottom of the tube. In fact, in a remarkable experiment that I still remember, he laid out these Petri plates on his bench, and we could see only SEC1 mutant cells at the bottom of this tube and all wild-type cells at the top. Well, this is a biochemist's way of doing genetics, which really greatly satisfied me. And so what he did next was to take a normal yeast culture, expose it to a chemical mutagen, incubate the cells at a restrictive temperature, and then ask for those cells that became dense at the high temperature by performing this buoyant density gradient. You punch a hole in the bottom of the tube, and you collect the densest 1% of the cells. You spread them out on a Petri plate, and you look at clones, and you pick the clones that are temperature sensitive for growth, and you evaluate whether these clones accumulate secretory enzymes inside the cell at the expense of secretion. And in another remarkable achievement of his, within the next year, he was able to identify hundreds of such mutations that map to 23 different genes, a pathway that we now know to be populated by probably over 100 genes, completely involved at all stages in the pathway. And let me run through some of the examples. In the initial collection, Novick identified about 10 genes, mutant alleles of which accumulate vesicles, as you saw previously, but he also found 2 other phenotypes. One mutant called SEC7, at a restrictive growth temperature, accumulates a structure that is not typically seen in a normal yeast cell. It's a Golgi apparatus, and you'd recognize that even if it didn't come with the letter G. This is a very clear elaboration of an organelle that must accumulate and halt traffic at the restrictive temperature. And sure enough, when these cells are returned to the permissive temperature, the Golgi structure dissipates and secretory proteins re-engage the pathway. Another phenotype that he saw at a restrictive temperature is the accumulation of membranes that are recognized as endoplasmic reticulum. Only in this case, the membranes are greatly expanded, distended, much larger than is otherwise seen in a normal cell, including the membrane that surrounds the nucleus, which is physically linked to the endoplasmic reticulum. So many such mutations defining different stages in the pathway and obviously identifying the crucial functions that control this pathway. Now, in surveying this very large set, we were struck after several years with the failure to identify mutations that interfere at the very first step in the pathway, the insertion of secretory proteins into the lumen of the ER. We would have predicted that such mutations would cause secretory proteins to build up in the cytosol and not to accumulate organelles, and we didn't find any such mutations. And so another few years went by, and another terrific graduate student, Ray Deshay, joined the group. Ray is shown here at one of our periodic banquets, sitting next to his wife, Linda Silvera, and so I can ask yet another successful couple to rise from the audience. Ray and Linda. Ray is now a professor at Caltech, and Linda's a professor at the University of Redlands. So let me tell you about what Ray did. Ray thought that one would need a more direct selection procedure to identify the genes that encode the subunits of the polypeptide translocation apparatus. And so he conceived of a quite brilliant selection scheme that I've summarized here. And so let me lead you through this scheme, and I hope you'll agree it really was very clever. Well, enzymes in the cytoplasm catalyze their chemical reactions because their substrates are present in the hydrophilic environment of the cytoplasm. One such enzyme is the enzyme that converts histidinol to its biosynthetic product, histidine, the amino acid. This enzyme, histidinol dehydrogenase, is a purely cytoplasmic protein having nothing to do with the secretory pathway. But Scott Emmer, when he was a postdoctoral fellow in my lab, found that if you append the sequence information that encodes a signal peptide, the signal peptide first discovered by Günter Blobel in the signal hypothesis, if you append the sequence information for a signal peptide to a cytoplasmic protein, you can force that cytoplasmic enzyme to enter into the lumen of the endoplasmic reticulum, a location in the cell where it can't do its chemistry. Ray showed that in this example as well, that if you append a signal peptide to the end information, to the end terminus of this enzyme, histidinol dehydrogenase, the enzyme now is dragged into the lumen of the ER, and it can no longer have access to its substrate, histidinol. And thus, these cells cannot manufacture histidine, and thus they cannot grow unless you provide histidine in the medium that can be taken up into the cells. So this is a setup for a genetic selection for mutations that cripple the translocation apparatus, because you'd expect that such mutations would leave at least some of this funny hybrid protein back in the cytoplasm, enough to catalyze the conversion of histidinol to histidine, thus to nourish cells that are themselves deficient in the production of histidine. Now, this, of course, is going to kill the cell if you cripple the translocation machinery completely, so Ray had to hit a happy medium to find mutations that would compromise this process at room temperature, but which would then kill the cell at a high temperature, 37 degrees. And the very first mutation that he identified by this procedure is a gene called SEC61, which we now know to be the translocation channel, the actual subunit of the channel that permits secretory proteins to enter the lumen of the endoplasmic reticulum. And I'll summarize a great deal of work in many laboratories shown here that SEC61 constitutes this channel in the endoplasmic reticulum in yeast. Tom Rappaport subsequently discovered the SEC61 protein as the channel in the ER in mammalian cells. And when we cloned the SEC61 gene in my lab, we found that its closest relative was an archaeal protein. In turn, that protein was closely related to a protein called SEC-Y, a protein that had been demonstrated by Ito and by Beckwith to be required for secretion, but which was shown very directly by Bill Wickner in his laboratory to be the translocation channel in E. coli. So we have in these genetic selections in yeast and in E. coli and biochemistry in mammalian cells a complete conservation of the translocation mechanism all the way from bacteria, archaea, into the eukaryotic kingdom. Well, let me summarize then not only Ray's work, but a great deal of work over many years that allowed us to define the genetic contour of the secretory pathway. Genes required for the translocation event, many genes required for a bidirectional flow of vesicles back and forth between the ER and Golgi apparatus, genes required for the organization of the Golgi apparatus and for the formation of transport vesicles, and in years subsequent to this, in the work of Scott Emmer and Tom Stevens, former postdoctorals in my lab, in their own labs at the University of Oregon and for Scott at Caltech initially, they discovered the genes that are required to sort proteins within the Golgi apparatus, separating proteins destined for the yeast vacuole from those that are destined for secretion. And finally, another set of genes required for the progression of mature granules to the cell surface. Now, at the time that we more or less completed these initial genetic studies, it became possible, using molecular cloning techniques, to identify the polypeptide nature of some of the SEC gene proteins. And the first and most important discovery on the nature of these proteins was made by Novick in his own laboratory, first at Yale, when he discovered that the SEC4 protein encodes a small RAS-like GTP binding protein, know to be required to target these vesicles to the cell surface. And we know from the many laboratories around the world that the so-called RAB proteins, roughly 3 dozen of them, are each required to target a vesicle to a different destination in the cell. So Novick then deserves the primary credit for having discovered this very important family of proteins. In my laboratory, we cloned several other genes, and we found no significant homology to anything that was known functionally. These were pioneer genes. Subsequently, we learned that homologs of these genes were to be found in the genomes of mammals, but even so, knowing that you have a homolog doesn't teach you what the function of that protein is. And so I began a quest to convince students and postdocs in the lab to attempt to establish a cell-free reaction, as you learned from Jim Rothman, one that could be used to isolate functional forms of the SEC proteins. So let me take you on a tour through the idea of how one can use genetics and biochemistry together to help solve a complicated problem. I learned as a university student at UCLA of the first example of this union of genetics and biochemistry through the work of Bill Wood and Bob Edgar, then young faculty members in the Division of Biology at Caltech. Here they are, shown here, doing an experiment together in the fall of 1965, the first year that Bill Wood was an assistant professor in that department. The fellow on the right is Bob Edgar. a pioneering geneticist who used traditional genetics to identify the structural genes required for the production of viruses, the bacterial virus T4. He was able to populate the genome of the T4 phage with mutations that cripple the production of virus at one point or another in its assembly to produce an infectious particle. Bill Wood came from a biochemical background. He was a graduate student with Paul Berg at Stanford, and of course, as we all were, was greatly influenced by the power of enzymology in the hands of Arthur Kornberg. And so the two decided to team up with one another in the fall of 1965 to do the following amazing experiment, which really resonated with my learning at the time. Here's their simple experiment. They would take bacteriophage having a mutation that would cripple the production of virus, and virus infected cells blocked at a different stage in the viral production. These two mutations would, if the bacterium was infected together with the two viruses, produce virus particles by what we know of as genetic complementation. But the question that Bill Wood posed is, could one produce biochemical complementations in a lysate of cells, a lysate where the two virus-infected cells were mixed? And as you see in this cartoon that was reproduced in Scientific American in 1966, virus particles were assembled. But more importantly, if you examine the data, you see at the outset this mixture had very few infectious virus particles. But over the course of a 30-minute incubation, an increase in 3 logs in the production of infectious virus particles. Really an astounding observation. This is the kind of experiment that makes a biochemist salivate because the signal-to-noise ratio is enormous. Motivated by that biochemical complementation, Bill Wickner and I used that logic to identify and purify proteins required for DNA replication in lysates of E. coli. And when I started my own lab in Berkeley, I was well aware of the possible use of genetics in this biochemical approach. But it always takes the right kind of person to do this project, and I was really blessed in the mid-1980s to have this fellow, David Baker, join the lab. David Baker is now an incredibly successful protein biophysicist in his own right at the University of Washington. But when he joined the lab, he succumbed to my persuasive influence to try to use lysates of yeast to reconstitute transport reactions. What he did was to establish, within the first few weeks of his experimental life as a graduate student, a gently prepared lysate of yeast spheroplasts that sustained what we imagine to be the first half of the secretory pathway in vitro. He could make a lysate, add a radioactive tracer of a secretory protein. That protein would enter the endoplasmic reticulum and progress successively through the Golgi apparatus. Quite a remarkable observation. His work was then complemented, crucially, by the work of Linda Hickey, who was pictured in a previous slide. Here's an experiment that Hickey did that, again, is one of the memorable experiments of my career. She used Baker's cell-free system to evaluate the transport in vitro of a radioactive tracer of a secretory protein in lysates containing membranes and cytosol prepared from one of the Sec mutants, Sec23. And so let's just examine the data. The open bars show the signal in the absence of cytosol, that is when membranes are incubated alone. The closed bars represent the transport activity observed at progressively higher temperatures, approaching the restrictive temperature when both membranes and cytosol are taken from a Sec23 temperature-sensitive mutant. Clearly, a temperature-sensitive defect in transport in vitro. And most importantly, if the cytosol is taken from a cell that has a Sec23 mutation, but which is complemented by a plasmid copy of the wild-type gene product, the lysates now are restored to temperature resistance for transport in vitro. So we were convinced, on the basis of this experiment, that Baker's in vitro system was a faithful replica of the physiologic event, dependent upon this, and turns out subsequently, on all the other proteins required for transport. Now this limb of the pathway, if you recall from the pathway slide that I showed, is populated by probably over 30 different genes. And though we were ambitious, we didn't want to have to purify all of those proteins at the same time in order to delve into a mechanistic question. And so at this moment, another crucial step of fate intervened with Michael Reksash, a terrific graduate student who joined the lab and who took Baker's reaction and simplified it to develop a reaction that allowed us to measure the budding of transport vesicles in vitro. At the same time, a wonderful postdoctoral fellow, Chris Kaiser, went back and had a closer look at intermediate steps in vivo between the ER and Golgi. And what Kaiser was able to show by a combination of morphology and genetic analysis is that the set of genes required for the flow of proteins between the ER and Golgi can actually be separated into two stages. Those required for the formation of vesicles, which showed extensive synthetic lethal genetic interactions, and a separate set of genes involved in the consumption of these vesicles by docking and fusion at the Golgi apparatus. At this point, all of these genes had been cloned and sequenced, so one knew the sequences, for instance, of these two genes. And at a very important point in Rothman and my career, Rothman's lab had cloned and sequenced the genes that encode his proteins that you've heard about, NSF and alpha SNAP, and they discovered that NSF encodes a protein, the gene encodes a protein that is very similar, highly similar to the Sec18 gene, and alpha SNAP encodes a protein that is very similar to our Sec17. So at this point, our two paths converged quite dramatically, and we were led to a grand synthesis that what he had been studying biochemically, we had also been studying genetically. Now let me return to Reksash, who simplified David Baker's reaction as is revealed in this very simple cartoon. What Reksash appreciated is that in the gentle lysate of yeast spheroplasts, membranes of the endoplasmic reticulum remain very large, so large that they will sediment when a test tube or a centrifuge tube is simply put in a microfuge and turned on and off. The membranes are large and they pellet very quickly. However, if these membranes are incubated with crude cytosol in the presence of nucleotide, small vesicles formed that could not be centrifuged in a microfuge, but which instead require sedimentation at high forces, such as in an ultracentrifuge, to pellet them. It became possible to use this really simple differential centrifugation assay to detect, ultimately to purify, functional forms of all of the proteins that are required for vesicle budding. And we discovered over the course of several years, including, crucially, these first experiments of Linda Hickey, that the Sec genes and proteins that Chris Kaiser showed to be required for budding in vivo are also required for the formation of transport vesicles in this Reksash assay. A wonderful postdoctoral fellow in the lab, Charles Barlow, took all of the purified proteins that we had obtained from this budding assay, mixed them together with isolated ER membranes, and we observed in our studies on a novel coat the formation of transport vesicles that at first glance look very similar to an image that Jim showed you of vesicles that contain a coat that he calls COP1, but molecularly we knew that this coat, though physically similar, actually is formed by a distinct set of gene products. Thus we call this coat COP2, and we spent many years studying how the COP2 coat engages cargo molecules and incorporates them into transport vesicles. But first, let me reintroduce a very important person that Jim has already mentioned, and that is Lelio Orci, with whom we had the pleasure of collaborating for many years. Orci, as Jim said, is or has been the master morphologist in the generation that followed George Palade. One would have to make the pilgrimage to Geneva to see him in his very elegantly outfitted office. To this day, after 20 years of collaboration, I still have yet to get Lelio to come to Berkeley to see me, but I have a trick up my sleeve because his son, married a woman who's from my neighborhood, and he will now be obliged to come to Berkeley to attend their wedding. So I think I'll finally get him back to California. Well, let me tell you, just in brief summary form, what we've learned about the COP2 proteins and how they assemble on the surface of a membrane and how they gather in and separate cargo molecules from other proteins that are not destined for transport. The molecules that we uncovered by this combination of genetics and biochemistry are shown here. One is a small GTPase called SAR1, a nucleotide exchange catalyst, an integral membrane protein in the ER called Sec12, and then 2 layers of a coat protein complex consisting of the products of 4 different Sec genes, 23, 24, 13, and 31, and then a random assortment of cargo molecules depicted here as green for those cargo molecules that are destined to be transported, and red for those molecules that are not and which will remain behind, forming the functional endoplasmic reticulum. So let me now show you this movie. If I can. Yes. So SAR1 in the cytoplasm collides with Sec12, extends an N-terminal amphipathic helix, embeds in the membrane, and recruits the Sec23-24 complex. As this diffuses within the plane of the membrane, it encounters different cargo molecules which engage binding sites on the Sec24 molecule, capturing cargo. The outer layer of the coat then begins to assemble, and with time, the coat actually polymerizes to form a cage that envelops the vesicle, enriched in cargo molecules. At the moment of fission from the ER membrane, SAR1 GTPase is activated by an intrinsic activating enzyme that's part of the coat. The coat is shed, and then the membrane of the vesicle exposes a SNARE protein that engages a cognate snare and a target membrane, and the vesicle is free to fuse with its target destination. Some specific events in the budding reaction have now been reconstituted, not only with pure proteins, but with synthetic bilayers. And I'll show you just 2 examples of that. We found, through the efforts of Marcus Lee, a wonderful postdoctoral fellow in the lab, that the very first step, the insertion of an activated form of SAR1, which extends an amphipathic N-terminal helix that embeds partway into the ER membrane, will by itself initiate the formation of a bud on the surface of a pure phospholipid bilayer, a synthetic membrane. So you see many examples here of buds now with time that elaborate into very long convoluted tubules extending from the surface of this pure phospholipid membrane. So we believe SAR1 initiates the budding event by creating a curvature on the membrane that then serves to recruit the other coat proteins. We found through the efforts of Ken Matsuoka and Eugene Futai that the further events in this process can be completed on addition of all of the other components and catalysts of this process. You see here an incubation with these membranes, but in the absence of the crucial nucleotide exchange catalyst for SAR1, on addition of that catalyst, one can now produce on the surface of synthetic membranes complete budded buds, and in other experiments, complete budded vesicles. So we have then fulfilled the requirements of the biochemist, but relied on the criteria of the geneticist to reconstitute this event. Work in other laboratories, principally Bill Balch, the fellow that Jim mentioned who was crucial in the development of the Rothman in vitro reaction, in his own laboratory discovered that the outer layer of the COP2 form creates a regular polyhedral cage that is the physical principle that bends the membrane to complete the budding process. We know also from Jonathan Goldberg the structure of all of the proteins at the atomic level, and we imagine that the Sec23-24 and SAR1 inner layer interacts at some point directly with the outer layer to allow cargo to be included as the bud forms and is cleaved from a membrane by fission. Now, in the course of our experiments, we were drawn away from yeast by the observation by clinicians of patient groups that have unique mutations in mammalian equivalents of the COP2 proteins. So let me show you just a little bit then about what we learned. All of these genes in yeast are encoded by single essential genes. That was crucial for our work because we were able to demonstrate that these mutations kill the cell. And yet all of these genes, as you see in this cartoon, are at least duplicated. For example, the GTPase SAR1 is duplicated. And we know from the work, including work conducted here in Stockholm, that this protein, SAR1b, seems to have a fairly unique role in the packaging of lipoproteins, crucially in the packaging of giant lipoproteins called chylomicrons at the endoplasmic reticulum. So here is a paper that appeared some years ago, a co-author, a colleague of yours, Urban Myrdal, where patients were found to have mutations, some of which severely cripple the B copy of the SAR1 protein. And these patients are seemingly normal, but they fail to secrete chylomicrons in the intestinal epithelium. So there was something really very fundamentally unique about SAR1b that distinguishes it from SAR1a, and we're exploring what that may be. Two other examples I'll leave you with affect the genes that encode the 2 forms of Sec23 and the 4 genes that encode the Sec24 subunits. First, let me describe Sec24. We know from our work in yeast and subsequently our work in mammalian cells that each of these Sec24 molecules is responsible for the recognition and discrimination of distinct cohorts of molecules that are captured into COP2 coats. We first showed this quite dramatically in a collaboration with David Ginty, who found a mouse mutant deficient in this protein, Sec24b. He found the mutant in a screen of mutants that have a defect in neural tube development, something that I wouldn't have imagined to be arrested fairly late in development, from my perspective, after embryonic development, by a mutation in a gene that we would imagine to be essentially essential for protein secretion. And yet, the Sec24b mutant allows development normally in the mouse embryo for 11.5 days and arrests at a stage similar to the defect that's found in the human disease spina bifida. And Devin Jensen in my lab was able to show that it's because Sec24b is fairly uniquely required for the packaging of certain cell surface receptors that must be displayed on the opposing surfaces of the neuronal epithelium in the early mouse embryo. More recently, David Ginsberg's lab found that mice deficient in Sec24a are otherwise normal. They grow quite normally, but have an unusually low level of cholesterol. And in some really quite creative experiments, Xiaowei Chen in his lab showed that Sec24a is responsible for packaging the very important secretory protein, PCSK9, in the liver. PCSK9 is a known regulator of the itinerary of the LDL receptor, and in the absence or severe reduction of the secretion of PCSK9, LDL receptor engages in a normal itinerary, which allows the cell to modulate the production of cholesterol and thus have a normal life, whatever that is for a mouse. So let me leave you with 2 examples here of the mutations that affect these 2 genes. Groups in Europe discovered a form of anemia, human anemia, which is attributable to lesions in Sec23b, and in collaboration with Simeon Boygiev, we've explored a mutation that affects this paralog, Sec23a. Sec23a, in a patient with a form of craniofacial dysmorphism, has a mutation in an invariant phenylalanine residue. The segments of the Sec23 gene are shown here. The 2 top lines are of the human paralogs. At the bottom is the yeast protein. They all have a phenylalanine residue. This residue in these patients is mutated conservatively to a leucine position, which has an unusual and very selective morphologic defect. which we explored, again in collaboration with Leila Orci, and were able to see a rather profound effect on secretion in fibroblasts harvested from one of the kids afflicted by this malady. So here, for instance, in an experiment conducted in Geneva, normal fibroblasts have this normal appearance of endoplasmic reticulum. Fibroblasts from one of the parents, a heterozygote with no pathology, seem fairly normal, but in the fibroblasts harvested from one of the kids, an enormous distortion of the ER, filled with granular staining material, more dramatically deficient even than the original Novick Sec23 mutation of yeast. We found subsequently that these cells are particularly deficient in the secretion of collagen, and we're exploring why that is. And finally, in looking in greater detail at the lesion in this Sec23 protein, we discovered biochemically that this mutation blocks the recruitment of the outer layer of the coat, and this was confirmed quite dramatically by Jonathan Goldberg at Sloan Kettering in New York in his crystal structure. So we now know that in these patients, particularly in cells that express Sec23A, budding is initiated but cannot be completed because the outer layer of the coat fails to be assembled. Well, let me leave you with what I consider to be the three most important lessons from the last 37 years of work in my laboratory at Berkeley. The major conclusions are as follows. Secretion and plasma membrane assembly are physically and functionally linked through a series of obligate organelle intermediates. We think from our original hypothesis that these vesicles are responsible for secretion and plasma membrane assembly, and the fact that we can arrest traffic and kill the cells demonstrates quite convincingly that these organelle intermediates are obligate stations on the path of secretion. By cloning and sequencing these genes and comparing them to their equivalent genes in higher eukaryotes, we can conclude that this pathway has been fundamentally conserved over 1 billion years of evolution. So once you invent a complex and efficient process, you want to reuse the parts as much as possible. And finally, our most recent experiments on COP2 have shown that this coat is responsible for discriminating cargo molecules, sorting them by the recognition of sorting signals on the membrane proteins, and it is also responsible for physically deforming the membrane to create a bud that includes these selected cargo molecules. Well, I'm grateful, of course, to the people I've introduced and whose pictures I've shown that were crucial over the path of my career at Berkeley, but I'm equally grateful to my current graduate students and postdoctoral fellows seen here on a special morning in October, early October, shown mocking my mustache by donning each of one themselves. Thank you for your attention.
So, thank you very much, Dr. Randy Scheckman. And I would now like to introduce our third laureate, Dr. Thomas Südhof. Dr. Südhof originally trained at the Georg August Universität in Göttingen, Germany, and received an MD and PhD there in 1982. In 1983, he moved as a postdoctoral fellow to the University of Texas Southwestern Medical Center in Dallas, where he started his laboratory in 1986. Südhof became an investigator at the Hughes Medical Institute in 1991 and was appointed professor of molecular and cellular physiology at Stanford University in 2008. Please join me in welcoming Dr. Südhof, who will present his Nobel lecture under the title, A Molecular Machine for Neurotransmitter Release.
Yeah.
No, it's fine. It's a true honor to be here. And I feel incredibly grateful to the committee for awarding this prize to me. It is also a tremendous pleasure for me to share this with Jim Rothman and Randy Scheckman. And as I will show you or tell you my approach to the same general questions that you've heard about is very different, but I believe highly complementary to what Jim and Randy have done. So let me start off by giving you some background. I'm a neuroscientist, and neuroscientists try to understand how the brain works. One way to look at how the brain works, possibly the most common way, is to think of the brain as a huge information processing machine that can be broken down into neural circuits. And a neural circuit consists basically of neurons that are connected by synapses. The synapses are the fundamental information processing units of the brain. They not only transfer information from one nerve cell to the next, they also process that information. Synapses differ dramatically in the brain. Synapses have special properties. They are plastic, but they all are governed by the same principle that was discovered by Bernard Katz many years ago, half a century ago, for which he was awarded a Nobel Prize in 1970. This principle is composed of three very simple steps. First, an action potential invades the presynaptic terminal. The action potential allows calcium channels to open, and the calcium then triggers neurotransmitter release rapidly. The released neurotransmitters finally binds to the postsynaptic membrane to elicit an electrical signal. How we study synaptic transmission is by using electrophysiology as a primary tool. What you see here is a typical trace where you see these blips. Each one of these is an event of fusion at the presynaptic plasma membrane that releases neurotransmitters to elicit postsynaptic responses. And as a result, we talk about excitatory and inhibitory postsynaptic currents depending on whether the signal inhibits or excites the postsynaptic neuron. What is remarkable about synaptic transmission, among others, is the unbelievable speed with which it occurs. Within a few milliseconds, and within these few milliseconds, everything happens from an action potential to the postsynaptic response. Of these three components of a synaptic transmission event, presynaptic calcium influx that triggers neurotransmitter release is arguably the most complex because it basically involves the transduction of a calcium signal into a membrane fusion event. The three basic processes that enable this rapid release of neurotransmitters that is triggered by calcium can be summarized as follows. First of all, neurotransmitter release requires fusion of membranes, specifically the fusion of synaptic vesicles with the presynaptic plasma membrane. Second, it requires the tight control of that fusion by calcium. Calcium acts incredibly fast, within a few hundred microseconds, which is about the same order of magnitude as gating of an ion channel. Calcium acts highly cooperatively in doing this, and it acts also in a very plastic manner. Finally, what is very important for synaptic transmission from a neuroscience perspective is that the calcium that flows in as a result of the action potential is highly localized. It flows in at the active zone right next to where the fusion happens, which is crucial for the tight coupling of an action potential to a neurotransmitter release reaction. So when I started my laboratory at UT Southwestern in 1986, neurotransmitter release fascinated me because of its importance, but also because of this inexplicable speed and precision. But at that point, not a single component of a synapse had been molecularly characterized. Now, 25 years later, liberally, a molecular framework that plausibly explains release in molecular terms has emerged. And this is what I'm going to talk about today. And that molecular framework is summarized here in a very cartoon fashion. And it composed of exactly the three different elements that I outlined to you before. There's a fusion machinery that mediates the fusion of the synaptic vesicles, composed of many of the same players that you've heard from Jim Rothman, and that basically constitutes the minimal fusion machinery. There is synaptotagmin that mediates the calcium triggering, and finally, there's a large protein complex that is partially shown here that forms the active zone of a synapse and that allows synaptic vesicles to be physically linked, among others, to calcium channels, thereby forming a relatively simple complex that puts everything together and allows the other two elements to function properly. And so what I will tell you today is a brief personal account of this machine, focusing on calcium. And let's start at the beginning. At the very beginning, when we started working on this, just like Jim, we were interested in how vesicles fuse. And in a paper that we published in 1993, we postulated a model for synaptic membrane fusion that was very primitive, but nevertheless, I think, quite accurate overall. And this model was based on 3 convergent observations, largely from other people. First of all, at that time, the laboratories of Cesare Montecucco and Reinhard Jahn, among others, had demonstrated in beautiful studies that clostridial neurotoxins, tetanus toxin and botulinum toxin, that block fusion at a synapse are enzymes, they're proteases that specifically cleave one of the 3 proteins referred to as SNARE proteins, synaptobrevin, also known as VAMP, SNAP25, and syntaxin. And this observation was seminal in our field because it really was the first demonstration that these proteins are involved in membrane fusion, and it sort of formed the baseline for much of what happened later. Second, in brilliant studies that Jim described, his laboratory demonstrated that the same 3 proteins that are cleaved by these clostridial neurotoxins form a complex with each other that he named the SNARE complex because they bind, there's the soluble NSF receptor. Soluble, oh, whatever. I always get tripped up at this. Jim can explain it much better. They're the receptors for the NSF and SNAPes that he discovered. And he showed, I think, in the most crucial observation, that the same proteins bind to each other in a single complex. And this complex thus represented a physical correlate, a biochemical correlate to the physiological observations by Montecucco and Jahn on the of these proteins as substrates for botulinum and tetanus toxin. And finally, my own laboratory, in biochemical experiments, observed that the SNARE proteins bound another protein that we named MUNC18. This protein, upon sequencing, was found to be homologous to MUNC18 and SEC1 genes that were discovered in beautiful work by Sidney Brenner's laboratory and Randy Scheckman's laboratory, respectively, as proteins that were essential for C. elegans movements and yeast secretion, respectively, although their mechanism of action was unknown. And when you put this together, the picture that we postulated based on this is that this together, based on these convergent observations, forms the minimal fusion machinery for a synaptic vesicle. In this view, MUNC18 is not a bystander, but a central action in the membrane fusion reaction. And this is based on the observation obtained later that MUNC18 is absolutely essential for vesicle fusion, whereas individual SNARE proteins are not. And similar observations were made in other intracellular fusion reactions. Just to show you the key experiment that was performed some years later by Matthias Verhache in my lab, what this demonstrates is that when you knock out MUNC18 in a mouse, you completely block any kind of neurotransmitter release. It becomes completely silent, the synapse. There's no presynaptic release, even though synapses form apparently normally, and postsynaptic receptors are normally present. A model for how MUNC18 functions in collaboration with SNARE proteins emerged from a lot of subsequent work, much of it from Jim Rothman's lab, as well as from the labs of many others. And this is summarized here in this cartoon, which suggests that prior to fusion, when synaptic vesicles are docked, the SNARE proteins are not yet engaged in any complex, and Munc-18 is bound to monomeric syntaxin in a closed conformation. What happens then is that there's a partial SNARE complex assembly that Jim already alluded to, which prepares the vesicle for fusion, and this partial SNARE complex assembly involves an opening of syntaxin, a rearrangement of the Munc-18 binding mode, and a partial zippering up of the SNARE complex. Subsequently, upon a calcium signal in the case of the synaptic vesicle fusion event, there is full assembly of trans-SNARE complexes with fusion per opening. This fusion per opening allows initial release of neurotransmitters, but is not sufficient for complete neurotransmitter release. The fusion needs to expand, pore needs to expand for complete neurotransmitter release out of the vesicle, and then you have the SNAREs and SM proteins assembled in cis complexes because now they're in the same membrane as opposed to different membranes in the trans complex. Finally, after all this has happened, in order for the whole machinery to be recycled and to be used again, as the chaperone NSF kicks in, and it dissociates the SNARE complex with the help of SNAPs, as Jim Rothman discovered, and allows synaptic vesicles to endocytose and recycle, and the SNAREs to be sorted again to their respective membranes. So this SNARE-SM protein cycle, in our view, underlies the fusion of synaptic vesicles as well as other intracellular SNARE-dependent fusion reactions. It is also important from a view of trying to understand disease. And I would like to give you two examples that have been particularly interesting to us. First of all, as it turns out, heterozygous de novo mutations, these are not homozygous, these are heterozygous and they're completely new, they happen in the germline, in Munc-18 are actually not infrequent. There's hundreds of cases, and they cause epileptic encephalopathy, a severe disorder that basically incapacitates the children completely, and demonstrating how important Munc-18 is. And indeed, in recent studies that I don't have time to discuss with you today, we created human neurons that replicate this heterozygous mutation and demonstrated that they do have an impairment in synaptic transmission, again, reinforcing the importance of Munc-18 or the SM proteins in this whole fusion process. Another example that I want to mention is that the assembly of SNARE complexes on this leg of this cycle here is actually chaperoned by non-essential but important proteins called CSPs for cysteine string proteins and synucleins. And this is potentially important for disease because, as you may know, alpha-synuclein aggregates accumulate in Parkinson's disease and in other neurodegenerative disorders. And in mice, loss of alpha-synuclein or CSP-alpha dramatically promotes neurodegeneration. And this might be important for understanding how neurodegeneration occurs in Parkinson's disease and other neurodegenerative disorders. Just to illustrate this, let me show you what happens in mice when you delete synucleins. What happens is basically, as shown here, that at P40, postnatal day 40, in these mice, biochemically, there's a relative loss of SNARE complex assembly, as shown here in yellow, compared to the middle-domain controls in black. And if you could study older mice at P200, that loss becomes quite dramatic. And these mice correspondingly develop an age-dependent neurological disorder that incapacitates them and eventually leads to premature mortality. Now, what's interesting here is that you can rescue this in cultured neurons by simply reintroducing synuclein in a very dose-dependent manner. What this slide demonstrates is that in these cultured neurons, this shows the assay, this is the summary graph, so let's focus on the summary graph. So in these cultured neurons, if you reintroduce alpha-synuclein in a dose-dependent manner, as a function of the alpha-synuclein levels, the SNARE complex assembly goes linearly up, as measured for all three SNARE proteins. And so this demonstrates that the SNARE alpha-synuclein can promote SNARE complex assembly, both in vivo and in a test tube, and that this may contribute to Parkinson's disease either by protecting against neurodegenerative insults or by promoting synaptic transmission. So let me move on and talk about the calcium triggering step, which is really what I've been most interested in my laboratory. At the same time as we were studying synaptic membrane fusion, we were systematically characterizing synaptic vesicle proteins with the idea that in order to understand how neurotransmitter works, the synaptic vesicle is a good starting point. And this approach led, among others, to the discovery of synaptotagmin, the calcium sensor for neurotransmitter release. In 1991, Reinhard Jahn and I published a review where we summarized what we had at that point accumulated in terms of an understanding of what a synaptic vesicle looks like. And what you see here is a cartoon from that review demonstrating the proteins that we had cloned at that point. Synaptobrevin was also cloned by Richard Scheller as VAMP, and the synapsins were originally purified in Paul Greengard's lab in beautiful work that dates back to the 70s. Among these proteins, what struck us most in the very beginning was synaptotagmin. And we were interested in this protein just simply based on its sequence, because it was an intrinsic membrane protein that had these motifs, which were called C2-domains. And these motifs were called C2-domains because they were homologous sequences that were highly similar to the second constant sequence that was previously observed in different isozymes of protein kinase C, but had no known function. Biochemical studies that were again carried out in collaboration with Reinhard Jahn and which were pioneered or driven primarily by Niels Brose, demonstrated that synaptotagmin bound calcium and that calcium binding to synaptotagmin promoted the binding of synaptotagmin to lipids, suggesting that synaptotagmin is a calcium binding protein. Moreover, Richard Scheller's laboratory, as well as our laboratory, demonstrated that synaptotagmin bound to SNAREs. And this altogether intrigued us tremendously because it raised us the questions of how does this protein bind calcium, because at that point C2-domains had known function, but the binding suggested that C2-domains may be calcium binding protein modules, and what is the physiological significance of this calcium binding? So we embarked on a long-term study to understand what happened, and much of it in close collaboration with structural biologists. And what we obtained was structures of both the C2A and C2B domains. What you see here are the structures that José Rizo obtained in his laboratory together with us over the last 15, 20 years. And what these findings showed is that C2 domains are compact, stable beta sandwiches that have flexible loops on top and the bottom. When we studied calcium binding to the C2 domains, we found that the calcium bound exclusively to the top loops and stabilized these loops, demonstrating that these are calcium binding modules that binds calcium via very specialized sequences in these top loops. And further, by physical studies on the exact sequences that bind these calcium ions, as shown here, demonstrated that they are closely opposed calcium binding sites that are coordinated by primarily aspartate residues in these two loops of the three tops loops of the C2 domains. Now this provided an explanation for how synaptotagmin binds calcium. And indeed, this type of calcium binding mode became prototypic for C2 domains in general that are found in many different proteins in the genome. However, it didn't tell us what this actually does. And so we asked whether the knockout of synaptotagmin 1 would selectively impair calcium-triggered release. And what we found that this was indeed the case. What you see here are electrophysiological tracings, EPSCs, that we obtained in collaboration with Chuck Stevens' lab using knockout mice that lacked synaptotagmin 1. And the key observation was that in wild-type neurons, there was this massive EPSC that was almost completely obliterated in the synaptotagmin 1 knockout mutants. So that demonstrated that synaptotagmin 1 is essential for fast calcium-triggered neurotransmitter release. As it turned out, it is not essential, however, for fusion, because when you do studies on the same synapses in which you stimulate fusion in these synapses by hypertonic sucrose, something that Bernard Katz had discovered stimulates secretion in a calcium-independent manner, we find that there's the same effect of sucrose on fusion in both the mutant and the wild-type synapses, demonstrating that fusion itself is not affected. So these studies demonstrated that synaptotacmin is only required for calcium-triggered fusion. And as a result, at that point, we came to the conclusions that synaptotacmin 1 is a synaptic vesicle calcium-binding protein that is essential for fast calcium-triggered release. In parallel, we found out that synaptotacmin does not act alone, that it needs an accomplice, a protein that we named Complexin. Complexin was discovered by Harvey McMahon in my laboratory as a protein that binds to the SNARE complex, hence its name. And following studies in many different laboratories, including our own, Niels Brose's and Christian Rosemund's, demonstrated that Complexin is an essential cofactor for synaptotacmin-triggered calcium-dependent fusion. In crystallography studies that we performed together with José Rizo, we found that Complexin binds to the SNARE complex by nestling into a groove in an anti-parallel manner, as shown here in this beautiful pink color on the background of this very colorful SNARE complex. There's been a lot of discussion that I won't go into today about what Complexin exactly does in terms of functioning as a clamp. But there is no doubt and general agreement that it functions as an activator of synaptotacmin 1 and that it is evolutionarily conserved. And I want to give you just one extremely recent example about what Complexin actually does. And this is based on my amazement when I looked at the sequences of the most early animals that you can identify in the animal kingdom, which are jellyfish, and whose genomes have been determined, whose genome sequences have been determined. When we looked at these sequences, we found that these animals that you would hardly recognize as animals, that really don't contain a classical nervous system, do encode in the genome multiple synaptotacmins and complexins. And so what we did in some experiments is we took Complexin from Anemostella vertensis and Nidarium as jellyfish and put it into mouse neurons. And when we did this, what we found, as shown here in this exemplary experiment, is here the sample traces, here's the summary graph, is that when we have a control neuron and we knock down complexins, you get a massive decrease in evoked EPSC amplitude, i.e. in calcium-triggered neurotransmitter release, because complexin acts as an activator. If you then put rat complexin back, you can rescue it. But you can also rescue it with a jellyfish complexin, and it just works just as well. So over hundreds of millions of time of evolution, this molecule retained its function, that you can basically put it back into a mouse neuron, and you can get activated calcium-activated reconstitution of calcium-activated neurotransmitter release. And that illustrates sort of the conservation and the enormous stringency of this machinery that involves synaptotacmin and complexin. So this is where we stood in about 1995, where we had identified synaptotacmin as a synaptic vesicle calcium-binding protein. We had shown it's essential for calcium-triggered release, and we had identified complexin as essential co-activator of synaptotacmin, although the function of complexin that I just described to you was really characterized mostly later. We stood in 1995, or sat some of us, as shown here in this picture, which was taken around that time, and key people who are involved in these studies that I described are shown here. And at that point, we had, together with others, identified the major components of the synaptic vesicle membrane fusion machinery and described the candidate calcium sensor for fusion. Crucial here was Yutaka Hata, who identified MONC18, Martin Geppert, who did all the synaptotacmin knockout studies, Harvey McMahon, who discovered, among others, complexin, Tai Li, who identified other synaptotacmins and showed that they bind to SNARE proteins, and others who are not shown, such as Mark Perrin, who cloned synaptotacmin, Niels Brose, who did all the initial work on synaptotacmin, and Bazbek Davletov, who really showed for the first time that a single C2 domain actually binds calcium. However, although at that point we felt quite sure that synaptotagmin is important and that SNARE and SM proteins do fusion, there were many issues that were not lingering in the background, that were actually very much upfront, which is that there was a lot of discussion of whether SNARE and SM proteins really do membrane fusion. Others suggested that synaptotagmin is a scaffold and doesn't act as a calcium sensor at all, and that its calcium binding is more sort of peripheral or incidental. And finally, we had no idea how calcium influx is localized to the site of vesicle fusion. And so what I want to do for the remainder of this presentation is focus completely on calcium and basically try to tell you about how we address these issues that came up during this time. And the first major question really was, does calcium binding to synaptotagmin really trigger fast release? Is synaptotagmin really the calcium sensor? And to address this question, we made use of our studies of the structure of the calcium binding site. As you can see here, these 3 calcium ions are ligated in close composition, coordinated in close proximity to each other by a number of conserved residues in these loops. And so we wanted to know if we could mutate some of these residues and not block calcium binding, but instead change the calcium affinity of synaptotagmin, and then test whether the same change in calcium affinity of synaptotagmin would translate into a change in calcium affinity of the release reaction. And so we designed mutations that shift the calcium affinity of synaptotagmin during SNARE or phospholipid binding, and these studies were carried out in close collaboration with Jose Rizzo, whose biophysical insights into how this might work were absolutely crucial in these studies. And what I want to tell you about are 2 particular mutations in residues that are right next to each other in this loop, in the C2A domain. This is the R233Q and the D232N mutation. So there are 2 different residues that change a positive and a negatively charged residue. And these 2 mutations dramatically have opposite effects. The D232 mutation, as shown here, strongly enhances the calcium-dependent binding of SNARE proteins to synaptotagmin, of synaptotagmin to SNARE proteins. What you see here is the calcium-dependent co-immunoposition of native brain synaptotagmin with SNARE complexes, and you can see here that as a function of the free calcium concentration, complexin co-immunoposition doesn't change, MUNC18 doesn't change, but the D232N mutant dramatically increases. So it's a gain of function mutation, how much SNAREs come down with the synaptotagmin. The other mutation, the R233Q mutation, does something very different. It actually affects relatively selectively the calcium-dependent binding of synaptotagmin to phospholipids. What you see here is an immunoblot of native brain synaptotagmin bound to liposomes as a function of the free calcium concentration. And what you see here is that this mutation shifts the apparent calcium affinity of synaptotagmin for phospholipids about 2-fold. There's about a 2-fold shift. And so we identified these mutations, we knocked them into mice, and we looked at what kind of effects do these mutations have on calcium-triggered neurotransmitter release. And what we found is that when we increase this calcium-dependent SNARE binding, we increase neurotransmitter release dramatically and approximately equivalently, and we increase the affinity, i.e. decrease the apparent KD of calcium. Conversely, when we decrease the calcium affinity of synaptotagmin for SNARE, of synaptotagmin for phospholipid binding, we decreased neurotransmitter release as a function of calcium, and we also decreased the calcium affinity approximately 2-fold, as shown here in an increase in the apparent KD. And so these studies formally proved that calcium binding to synaptotagmin is what actually triggers neurotransmitter release, that synaptotagmin is really the calcium sensor. And so we concluded that calcium binding to synaptotagmin 1 triggers fast release. So that was great, but there were other problems. One problem was that synaptotagmin is expressed in many forms that are highly homologous in the mammalian genome, actually 16. Of these 16 synaptotagmins, 8 bind calcium and 8 don't. So how is it possible that we have 8 synaptotagmins that bind calcium, which come in 2 different colors, as shown here? How is it possible that we have these synaptotagmins and still have a phenotype when we knock out synaptotagmin 1? What do these synaptotagmins do? Do they all do different things? What is interesting about these 8 synaptotagments is that 4 of them are monomers, whereas the other 4 are covalent dimers. And this is meant to be illustrated here in these 2 colors. But otherwise, they're very, very similar. So we asked which ones of these 8 synaptotagments can actually function as calcium sensors for fast neurotransmitter release. And what we found in systematic studies where we reintroduced the 8 different synaptotagments into knockout neurons from synaptotagment 1 that lack fast calcium-triggered neurotransmitter release, that synaptotagment 1 obviously rescues, synaptotagment 2 rescues, and synaptotagment 9 rescues. So 3 of the 8 can do it. So that is great. But what is puzzling is that synaptotagment 7, which is the 4th blue synaptotagment that I showed you earlier, cannot rescue. So synaptotagment 1, 2, and 9 selectively rescue fast release in SID1 knockout neurons, but with distinct properties that I won't discuss today, whereas synaptotagment 7 does not rescue. So this leads to 2 new issues. If you look at these blue synaptotagments, if they can sort of substitute for each other, why does the synaptotagment 1 knockout have a phenotype if SID2 and SID9 can compensate? And why does synaptotagment 7 not function in release if it's so similar and has basically the same structure? So to address these issues, we analyzed recently at the single cell level the expression of synaptotagments in isolated, patched hippocampal neurons. And what you see here is an analysis at the single cell level by quantitative RT-PCR that is shown here in this heat map, where red means a lot and blue means nothing. And what you see when you go through this here and just focusing only on the synaptotagments that are important, that in these neurons from the hippocampus, SID2 and SID9 is blue. So that means there's no SID2 and SID9, which explains why there is a phenotype. And further studies over the years have demonstrated that these different synaptotagments are differentially expressed in different types of neurons, which also correlates with the fact that they have slightly different properties, that they confer on these neurons. What you can also see, however, is that SID7 is highly expressed with SID1. In fact, its levels are on average higher than those of SID1. So this raised another issue. What does SID7 do? If it's so similar, why doesn't it work just like the others? And the answer to this question came out only very recently, where we went back to our initial observation shown here, that when you knock out synaptotagment 1, you block most fast-triggered release. But if you look closely, there's actually some release left here. There is not all release gone. In fact, the observation that there was some release left led some in the field to suggest early on that synaptotagment is not the calcium sensor, because if it were the calcium sensor, these people argued, there shouldn't be any release left. So we wondered whether this little bit of release that's left here may be actually due to synaptotagment 7 that is expressed in such high levels. And so what we did is, in recent studies, we used knockout and knockdown approaches to analyze this. And what we found, as shown here, is that in a controlled synaptotagment 1 knockout, there's a certain amount of residual release left in response to a single action potential. This is unchanged if you simply overexpress synaptotagment 7 wild type, which we showed earlier can't rescue the loss of synchronous fast release. But if we knock down synaptotagment 7, as shown here, you lose virtually all of whatever is left here on this remaining release. And that can be rescued by reintroducing wild-type synaptotagment 7, but not by reintroducing mutant synaptotagment 7. And so these studies, I think, establish that all 4 synaptotagments that are in this blue class here are involved in neurotransmitter release, and they have different properties, and they account for virtually all of the calcium-triggered neurotransmitter release. Different properties are able to confer on neurons special changes or special properties in terms of their network abilities, but in principle, they all operate by the same mechanisms, and they all mediate the same reactions. Moreover, what I haven't shown you is that the same blue synaptotagments not only work in synaptic neurotransmitter release, they actually operate in many different other types of calcium-triggered fusion. For example, hormone secretion. mast cell degranulation, and so on. So this is a general principle in biology. So then the remaining question is, what do the red synaptogments do? Why are they existent? Why are there 4 of these red isoforms? And I'm only going to show you some data on synaptogment 10 that illustrates that the same paradigm that I demonstrated to you for the blue synaptogments also applies for the red synaptogments. And what this data that we published a couple of years ago demonstrates is that synaptogment 10 is highly expressed in olfactory bulb neurons where it co-localizes with IGF-1, a growth factor, as shown here in this double immunofluorescence labeling. When we looked at whether or not synaptogment 10 is essential for the calcium-dependent secretion of IGF-1, we found that a knockout of synaptogment 10, as shown here, compare the control with the dark red one, causes a massive impairment of calcium-triggered IGF-1 secretion that can be rescued with wild-type synaptogment 10, but not with mutant synaptogment 10 that has no functional calcium-binding sites. Synaptogment 1 knockdown has no effect on IGF-1 secretion, demonstrating that synaptogment 10 is essential for the calcium-triggered release of IGF-1 in these neurons, and moreover, that this involves calcium-binding sites, intact calcium-binding sites in synaptogment 10. Loss of IGF-1 secretion in these neurons decreases the size of the neurons, and as a result, there's fewer synapses. And this can be rescued with IGF-1. To illustrate this to you, let me show you that when you use the same synaptogment 10 knockout neurons, and you look at the total synaptic response, it's massively decreased. And that's not because synaptic transmission is impaired, it is simply because there's fewer synapses, because the neurons are smaller, as illustrated by the fact that the capacitance is significantly decreased as well. Simple addition of IGF-1 to the medium completely rescues this phenotype, demonstrating that the entire phenotype is only due to a loss of IGF-1 and nothing else. So synaptogment 10 is a calcium sensor for IGF-1 exocytosis. All of the blue synaptogments require complexin for activation. Is the same true for the red synaptogments? To ask this question, we looked at IGF-1 secretion as a function of complexin. And when we knock down complexin, as shown here, we severely impair IGF-1, calcium-triggered IGF-1 secretion. This is as a function of calcium. And this can be rescued by reintroducing wild-type complexin, demonstrating that IGF-1 secretion that is mediated by calcium binding to synaptogment 10 also involves complexin. So these findings have general implications, I believe, for both neuroscience and cell biology. And this is summarized in this slide here, which shows in a very schematic manner that in a neuron, there can be many different pathways of calcium-dependent exocytosis. In this case, there's neuropeptide vesicles that have classical neuropeptides and that contain the blue synaptogments, as shown here. There's IGF-1 containing vesicles that contain IGF-1 and have synaptogment 10, which is not shown in red, but should have been shown in red. And there's synaptic vesicles, which again contain the blue synaptogments. These 3 pathways are independently sorted and independently regulated. We believe that different synaptogments impair on these different types of vesicles different properties, and thus, even in a single cell, a calcium signal can be translated depending on its dynamics and amplitude into different secretory responses that are governed by different synaptogments. Nevertheless, in every case, complexin is a cofactor that is required for the activation of the fusion reaction together with synaptogment. So diverse, non-redundant synaptogments use the same complexin-dependent mechanisms for different calcium-dependent membrane fusion reactions. So to summarize, what I've shown you is that synaptogment is a synaptic vesicle calcium-binding protein that is essential for calcium-triggered release, that uses complexin as an essential cofactor, that calcium binding to synaptogment 1 triggers fast release, and that other synaptogments perform analogous functions in calcium-triggered release with complexin as a cofactor. And this basically, I think, accounts for most of the properties, of these amazing properties of fast calcium-triggered neurotransmitter release. However, it still leaves open the last question, which I'll address in my remaining 5 minutes, which is, how about calcium influx? How is calcium influx located specifically here? And what happens if you don't have localized calcium influx at the active zone? Action potentials and release become uncoupled, and release becomes desynchronized and decelerated, and synapses basically don't work. And the importance of the localized calcium influx cannot be overestimated. Like in real estate, location is everything. And what I'm going to tell you is that there's a protein complex that accounts not only for the localization of calcium channels right next to darked vesicles, but mediates actually the very darking of these vesicles to the same protein complex. And moreover, this protein complexes do other things at the synapses as well. And this protein complex contains as a central component RIM, a protein that we initially cloned because it binds to RAP3, a GTP binding protein on the synaptic vesicle, but turns out to be the central sort of key to everything that happens in the active zone. When you knock out RIM, not surprisingly, you completely impair neurotransmitter release at both excitatory and inhibitory synapses. This impairment leaves some neurotransmitter intact, but involves many different changes in the synapses, including a loss of docking of vesicles and a loss of priming of synaptic SNARE complexes. It also likely involves a change in calcium influx, as was shown in initial experiments, because in the remaining release in these synapses becomes highly desynchronized and decelerated. The rise times go way up, and the standard deviation of the rise times, which is a desynchronization index, increases even more, suggestive that calcium influx has lost its localization. And indeed, consistent with this interpretation, if you introduce a slow calcium buffer that normally has very little effect, as shown here, on neurotransmitter release into the double knockout neurons, as shown here, you get a massive impairment of the normalized iPSC amplitude that is much bigger than that in controlled synapses, suggesting that calcium has to be around longer until it can actually trigger neurotransmitter release. And so inspired by these findings, Pascal Kayser, who was at that point a postdoc in my laboratory, measured calcium influx directly in RIM-deficient neurons. And these experiments were done by patching a neuron in culture and filling it with a tracer dye and a calcium indicator dye. And then Pascal measured in individual presynaptic terminals the calcium signal as a function of an isolated action potential. So you can actually measure the calcium signal for each individual action potential, as shown here. And what these experiments demonstrated, that in a control condition, there is a massive calcium influx, as shown here, which can also be plotted as a cumulative probability diagram, as shown here as the peak amplitude. When you knock out RIM, this influx dramatically decreases, and this probability plot shifts to the left. Calcium influx is severely impaired. And this can be rescued with a fragment of RIM that we had shown binds to the calcium channel directly, and that rescues completely, as shown here in red, the phenotype. These experiments, as well as many other experiments, demonstrated that RIM specifically acts at the presynaptic terminal and not at the dendrites, because there's no phenotype there, to mediate the localization of calcium channels right next to the vesicle, as well as allowing vesicles to dark and mediating the priming of SNARE complexes for calcium-triggered fusion. So to summarize, RIM is the essential glue in this, and it explains, together with its binding partner RIM-BP, MUNC13, and others, how this orchestration occurs, which allows a presynaptic terminal to couple calcium channels and calcium influx to dark vesicles, the SNARE machinery, and synaptotagmin. So in conclusion, what I've tried to tell you is that vesicles, synaptic vesicles, are exocytosed as a function of calcium by a fusion reaction that is catalyzed by SNARE and SM proteins that form a complex between the two membranes that are supposed to fuse. That this involves synaptotagmin as a calcium sensor, which acts as a function of calcium directly on the SNARE complex and on the phospholipids, and that finally, localized calcium influx is enabled by a supramolecular complex in the simplest possible manner, which directly links the vesicle to the calcium channel and thereby ensures a nanodomain coupling between the calcium channel, synaptotagmin, and the fusion machinery. And so functionally, the fusion, calcium triggering, and active zone complexes form a single interacting nanomachine that together fast neurotransmitter release. And that explains these magical properties that a synapse exhibits in terms of speed and plasticity. In closing, I would like to acknowledge the people who have really allowed me to do this. And because, you know, I didn't do much of the work, it's others who do the work. And I would like to first acknowledge my mentors, Victor Whitaker, who supervised my thesis work, and Mike Brown and Joe Goldstein, who were really in every respect, my role models, and who taught me how to do science. Most of the work that I discussed with you was carried out at UT Southwestern, where I spent many, many productive and very enjoyable years until I moved to Stanford 5 years ago, which has also turned out to be a wonderful place to work. I've been fortunate to have really incredible collaborators over the years, and I have to say one of the biggest joys in science is to collaborate with people. It is a tremendous, very rewarding to actually develop ideas together with somebody else. And so it started off with collaborations, long-term collaboration with Reinhard Jahn and Jose Rizzo, Bob Hammer, Rob Malenka, and many others. And finally, as I already alluded to, the work that I described to you was largely carried out by students and postdocs in the lab who were absolutely instrumental. I mentioned some of them already. Here is a more complete list. Without going into too much detail, I just want to point out that without the dedication of the people who I work with, or who work with me, or who work for me, or whatever, doing science would not be any fun and would be completely unproductive. And I think that my collaborators, my former students and postdocs, most of whom have gone on to do fascinating work on their own, really deserve an enormous amount of credit for everything. Thank you very much for your attention.
While thanking the three laureates for having delivered exciting, thoughtful, inspirational, energizing, and most memorable talks, I would like to invite James, Randy, and Tom to join me here on the stage for allowing us all, the whole audience, to salute you by a final round of applause. Please. We have now reached the end. There will be a brief photo session involving the laureates, and I now leave the floor to the head usher, who will provide some critically important pieces of housekeeping information.
Thank you so much, everyone, for coming today. It's great to see that it doesn't matter how big a lecture hall Karolinska builds, there still aren't enough chairs for everyone who wants to come and listen to these lectures. In order to make it easier for us to leave the room in an orderly fashion, I would like that the guests of the Nobel laureates and the members of the Nobel Assembly please use the door that's up at the top corner of the room there.